Somashekar, your botany teacher. Let me know in the comments how your NEET exam went. I just saw the paper and I'm overjoyed. I'm pretty sure all of you must have done really well because as always, it has been proven again that NCERT is the king. You call it the Bhagavad Gita, the Quran, the Bible, whatever it is, but NCERT is the king. Every question in the botany section was from NCERT itself, right? Let me know in the comment section if I'm clearly visible and audible. I'm asking this a little too late, but just let me know. I don't see any comments yet. Is anybody there? Please let me know in the comments. I'm not clearly able to see the comments yet. Wow, Nathan, that's really nice to hear. Congratulations. It feels so good, right? After all the effort that you've put in for 2 or 2+ years and you finally see a paper like this and you get like happy tears in your eyes. Hi Arpit. Very nice paper. Mario is saying he or she is going to get 715. That's brilliant. Congratulations. All right, let's get started with solving the paper. Could you please increase the font size of the comments? I'm not able to clearly see it. Ram sir? Just give me 1 minute. There's some technical thing that we need to take care of. All right, great. 580. Arpit is expecting 580. Physics में एक wrong हो गया. It's okay, happens sometimes. Nazim, you're upset. Why? It was such an easy paper. At least for botany, I can speak. I don't know about the other subjects. All right, without wasting any more time, let's discuss NEET 2024 botany section. Is everyone ready? Give me a thumbs up and let me know in the comments. Brilliant, Shrivani. Congratulations. Let's get started. Here is the first question. The paper or the code that we're going to be solving today is T3 English. You may have noticed how so many questions the concepts that were asked in the previous year, similar type of questions that appeared in this year's NEET as well, right? Oh, brilliant, Mario. All right, let's get started then. Here is our first question. This is from the chapter Sexual Reproduction in Flowering Plants based on pollination. It is a statement-based question. There are 5 statements given. We are required to identify which the correct statements are. Statement A reads, the flowers of Vallisneria are colorful and produce nectar. I'm sure you all know that Vallisneria is pollinated by water. It's an aquatic plant that's pollinated by water. Since it is pollinated by an abiotic agent, there's no need for flowers of that kind to be colorful or produce nectar. So that option clearly is, that statement clearly is incorrect. Statement B reads, the flowers of water lily are not pollinated by water. Water lily for sure is an aquatic plant, but since the flowers are produced above the level of the water, it is pollinated either by wind or by insects. Despite being an aquatic plant, pollination does not happen by the agency of water here. So this is the correct statement. In most of water pollinated species, the pollen grains are protected from wetting. Yes, that is the sentence as it is picked up from NCERT. This also is true. Pollen grains of some hydrophytes are long and ribbon-like. Yes, that is also correct. In some hydrophytes, the pollen grains are carried passively inside water. That also is correct. In the question, we are asked to identify the correct statements. Statements B, C, D and E are correct. So the correct answer to this question is option 2, right? Wow, Gangadhar, you're expecting 720 marks. Brilliant. Congratulations. Next question. This is a question from the chapter Biodiversity and Conservation. The type of conservation in which threatened species are taken out from their natural habitat and placed in a special setting where they can be protected and given special care are called. To be more specific, this type of conservation is called ex situ conservation, but it's not mentioned in the options. It's not a semi-conservative method. It has nothing to do with sustainable development. It's clearly not in situ conservation. So the closest answer that we have here is biodiversity conservation. It is biodiversity conservation. To be more specific, it is ex situ conservation, but it's not mentioned in the options. So let's mark. So according to me, 4 is the correct answer. Alright, now let's take a look at the next question. This is a question from the chapter Biomolecules. Biomolecules is generally taught under the zoology syllabus, but questions from biomolecules appear both in the botany as well as the zoology section. So here is a question from the chapter Biomolecules in the botany section. Let's take a look at it. Inhibition of succinate dehydrogenase enzyme by malonate is a classical example of. This is regarding the various factors that affect the rate of enzyme activity. So this is an example of competitive inhibition. So the correct answer here is option 1. Has everyone marked this answer correctly? Let me know. The correct answer here is competitive inhibition. What happens in competitive inhibition? The substrate and the inhibitor are similar in structure. They both compete for the same active site. So this is an example for competitive inhibition. Okay. Next question. This is a diagram based question from the chapter Sexual Reproduction in Flowering Plants. The diagram that we have here is of a section of a monocot seed. What they've asked us here is to identify the part of the seed from the given figure which is destined to form root when the seed germinates. So which part of this seed is going to form the root? This is a monocot root. In the monocot root, you have the endosperm here and this portion is the embryo. In the embryo, you have one cotyledon called the scutellum, and in the embryonal axis, you will have the radicle, the plumule, and then you'll have the coleoptile and coleorhiza. Over here, D represents coleorhiza, C represents the radicle, B I think is your plumule, and A is coleoptile. Everybody knows that the root system arises from the radicle. In this diagram, radicle is represented by letter C. So the correct answer to this question is option 1, right? Okay, let's move on and take a look at our next question from the chapter Anatomy of Flowering Plants. In Anatomy of Flowering Plants, specifically this question is from the TS of isobilateral leaf or monocot leaf. The question reads, bulliform cells are responsible for. Basically, they're asking for what is the function of bulliform cells. You know that these cells are present in the adaxial epidermis in monocot leaves like grasses, and whenever there is water stress for the plant, these bulliform cells, which are usually large, colorless and empty, will lose water. They will become flaccid and result in curling of the leaves. When these leaves curl, there is less exposure to sunlight, and as a result, the rate of transpiration will be reduced, right? So the correct answer to this question is option 3. It does not help in increasing photosynthesis in monocots. It does not provide large spaces for storage of sugars. It causes inward curling of leaves in monocots. That is the correct answer. Okay. So it does not protect the plant from salt stress. It protects the plant from water stress. Correct answer here is option 3. Let's take a look at our next question, which is from the chapter Photosynthesis in Higher Plants. Which of the following are required for the dark reaction of photosynthesis? A. Light, B. Chlorophyll, C. Carbon dioxide, D. ATP, E. NADPH. We all know that photosynthesis happens in two phases. There's a light dependent phase or the light reaction, and there's the light independent phase or the dark reaction. In the question, they've asked us among these given list of things, what are required for the dark reaction? Dark reaction does not require light, but it requires the products of the light reaction, right? So light is not necessary for dark reaction. Chlorophyll is not necessary for dark reaction. Both of these are used in your light reaction. In the light reaction of photosynthesis, the pigments like chlorophyll will trap the energy that is present in the sunlight and convert that or use it to make energy rich molecules like ATP and NADPH, which the cells will further use later in the dark reaction to fix carbon dioxide. So in the dark reaction, carbon dioxide, ATP and NADPH are used. So the correct answer to this question is C, D and E only, which is option 1. Alright. Now let's take a look at our next question. Formation of interfascicular cambium from fully developed parenchyma cells is an example for. Interfascicular cambium forms during secondary growth in dicot stems. So what basically happens during the formation of interfascicular cambium is parenchyma, which is a permanent tissue. Since it is a permanent tissue, it would have lost the ability to divide. Cell division is not possible by parenchyma cells. But this differentiated parenchyma tissue, which has lost the ability to divide, will regain the ability to divide and become meristematic. And that meristematic tissue is what we refer to as interfascicular cambium. Whenever a fully differentiated tissue that has lost the ability to divide regains the ability to divide and becomes meristematic, what do we call it? We call it de-differentiation, right? We've learnt about it in the chapter Plant Growth and Development. De-differentiation is when a fully mature permanent tissue goes back to becoming meristematic. So the correct answer to this question here is option 1, de-differentiation. Some of you may have gotten confused in this question between de-differentiation and redifferentiation. Whenever meristematic activity is regained, we call it de-differentiation. Alright, now let's take a look at our next question from the chapter Biotechnology. Generally, you learn biotechnology under the zoology syllabus, but in the NEET exam, from biotech chapters, questions are asked in both botany and zoology sections. This is one such question from biotech chapter in your botany section. So, HIND2, that is the name of a restriction endonuclease, always cuts DNA molecule at a particular point called recognition sequence, and it consists of how many base pairs? We all know that restriction endonucleases specifically cleave DNA, cut the DNA, make what do you call it? Cleave, yeah, the phosphodiester bonds in DNA and cut DNA, right? So there are specific sequences that these enzymes will recognize. They will bind to those specific sequences and cut the DNA. So for this HIND2 restriction endonuclease, the length of the recognition sequence is 6 base pairs. Okay, so this is a memory based question. This is a statement that was as it is in your NCERT textbooks, right? So the correct answer here is option 4. All right. Now let's move on and take a look at our next question. This is a question from biodiversity and conservation. In biodiversity and conservation, you've learnt about how biodiversity is not uniform throughout the world. There are certain regions where biodiversity is high, species diversity is more, and there are certain regions on Earth where species diversity is less. We've all learnt about the latitudinal gradients. On Earth, towards the tropics, equator has the highest species diversity. As you move towards the poles, on both sides, northern side and the southern side, species diversity will keep on decreasing. And there are 3 reasons that we've learnt about which causes a decrease in species diversity as you move from the equator to the poles, right? So this is a question based on that concept. The question reads, tropical regions show greatest levels of species richness because. They are asking for what are the reasons why there is highest species diversity or highest species richness in the tropics. Let's read the statements one by one. The first statement is, tropical latitudes have remained relatively undisturbed for millions of years. Hence, more time was available for species diversification. This is a statement as it is from NCERT and this is a true statement, right? What does statement B say? Tropical environments are more seasonal. That is not correct. Tropical environments are not more seasonal. It is the temperate environments that are relatively more seasonal. What does option C say? More solar, not option C, statement C. More solar energy is available in the tropics. That is correct. Constant environments promote niche specialization. That is also true. Tropical environments are constant and predictable. That is also true. So among the given 5 statements, only one statement is incorrect, and that is statement B. So the correct statements are A, C, D and E, which is option 3. Right, Mr. Famous says it's 3. It is 3 indeed. Let's take a look at our next question. Which one of the following is not a criterion for classification of fungi? This is a question from the chapter Biological Classification. In this chapter, you've learnt about different kingdoms, Monera, Protista, fungi, and all of that. So, you've also learnt about the detailed classification of fungi. There is a mention of the criteria used for classifying fungi, right? So, among the given options, which is not a criterion that is used for classifying fungi? That is what we are required to identify. Mode of spore formation, yes, that is a criterion that we use. Fruiting body, morphology of mycelium, whether it is septate, aseptate, that is a criterion used for the classification of fungi. What is not a criterion for classification of fungi is its mode of nutrition. So the correct answer to this question is option 4. In all fungi, regardless of which class it belongs to, regardless of which group it belongs to, mode of nutrition is the same. They all show heterotrophy, more specifically absorptive heterotrophy. Right? So, option 4 is the correct answer to this question. Now, let's take a look at our next question. This is a question from the chapter Photosynthesis in Higher Plants. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in the Calvin cycle? What happens in the Calvin cycle? We have 3 steps. There is carboxylation, there is reduction, and there is regeneration. In the reduction step, per carbon dioxide, you require 2 ATP and 2 NADPH. And in the regeneration step, per carbon dioxide, we require 1 ATP. So, in total, per carbon dioxide, we require 3 ATPs and 2 NADPH, right? So, in reduction step, we will need 2 ATPs and 2 NADPH molecules. And in the regeneration step, per carbon dioxide, we will require 1 ATP molecule. So, totally per carbon dioxide, we will need 3 ATP and 2 NADPH. So, the second option is correct. Option 2 is the correct answer. Alright, now let's take a look at our next question. This is a question from the chapter Biodiversity and Conservation. Let's read the question. These are regarded as major causes of biodiversity loss. There are 4 major causes for biodiversity loss. We call it the evil quartet. Among the given list of phenomena, which are the ones that cause biodiversity loss, that is what we require to identify. So over here, there is overexploitation, co-extinction, mutation, habitat loss and fragmentation, and migration. Overexploitation sure causes loss of biodiversity. We've learnt about examples of passenger's pigeon and stellar sea cow and all of that, right? So overexploitation definitely causes a loss of biodiversity. Co-extinction, yes, this also is a major cause of biodiversity loss. Mutation does not cause loss of biodiversity. Habitat loss and fragmentation, this is the major cause for biodiversity loss, and migration also does not cause loss of biodiversity. So among what is given as the evil quartet, overexploitation, co-extinction and habitat loss and fragmentation. These are the processes that will result in a loss of biodiversity. So A, B, D. Option 2 is the correct answer. Right? Now, let's take a look at the next question. This is a question from the tissue culture part, which is included in your biotechnology chapter. Right? So, let's take a look at the question. The capacity to generate a whole plant from any cell of the plant is called differentiation, somatic hybridization, totipotency, micropropagation. So, what happens here? You know, when we in tissue culture, if you want to generate many plants with one single plant, if you want to get similar kind of plants without any variation, you can take any living cell from the plant, use that cell or that tissue to generate new plants, right? This is possible in plants because in plants, every single cell that is living has the ability to give rise to an entire organism. So, what do we call that? What do we call that ability or the capacity to generate a whole plant from any cell of the plant? What do we call that? We call it totipotency. So, the correct answer to this question is option 3. Hi Vinayak, good evening. It will be the same questions, but it may not be in the same order. Okay. Let's look at our next question here. This is a question from Organisms and Populations chapter. They have given us an equation of the logistic growth curve, which is also called Verhulst-Pearl logistic growth. The equation is dn by dt is equal to Rn multiplied by k minus n divided by k. What they are asking us here is, what does k in this equation indicate? What does k indicate here? K is the carrying capacity. Now, what is the carrying capacity? Whenever a habitat or a geographical area has limited resources, any population cannot grow indefinitely over there. That area, that habitat can only support a maximum number of individuals, right? So that maximum number of individuals that a habitat can support, whenever that habitat has limited resources, that is what we call the carrying capacity. Okay, so the correct answer here is option 1. Now let's look at our next question. This is a question from the chapter Cell Cycle and Cell Division. The question reads, spindle fibers attached to kinetochores of chromosomes during. In the options, all the phases of karyokinesis of mitosis are listed. You have anaphase, telophase, prophase and metaphase. We are required to identify in which phase exactly spindle fibers attached to kinetochores. Now, what are kinetochores? Kinetochores are these proteinaceous structures, disc-like structures that are present on either sides of your centromeres in chromosomes, right? So, if this is your chromosome, this is your centromere. On either sides of the centromere, you will have kinetochores. So, these kinetochores will attach to the spindle fibers when? They attach to the spindle fibers in the metaphase. In metaphase, whenever the chromosomes are fully condensed, they come and form a line at the center of the cell, which we call the equatorial plate or the metaphase plate. It is in that phase that the spindle fibers will come and attach to the kinetochore, right? So, the correct answer to this question is option 4. Now, let's take a look at the next question. This is a question from the chapter Morphology of Flowering Plants. They have given us 2 diagrams of flowers. These diagrams are taken from NCERT itself. Let's see what this question is asking us. Identify the type of flowers based on the position of calyx, corolla and androecium with respect to the ovary from the given figures A and B. As you can see, this is A and this is B. We've learned about many different ways of classifying flowers. One of the ways in which we can classify flowers is based on how the other whorls are arranged, like the calyx, the corolla and the androecium are arranged with respect to the gynoecium. Okay, so the 3 different types of flowers that we can find, if that is the criterion for classification, are your epigynous flowers, your hypogynous flowers and the perigynous flowers. So whatever diagrams are given to us here in this question, both of them, you can see how the other whorls, this is your gynoecium, this is your gynoecium here, you can see how the other whorls are arising at a similar level. If the other whorls are arising at a similar level as that of the gynoecium, we call it perigynous flowers. Okay, peri means around. Around the gynoecium, at pretty much the same level, the other whorls are arising, we call it perigynous flowers. Okay, so over here, both A and B are perigynous flowers. So the correct answer would be option 2. Alright, now let's look at our next question. It is a match the following type of question from the chapter Biological Classification. Again, this is from fungi, kingdom fungi. We saw another question related to kingdom fungi. This again is another question related to kingdom fungi. Here they've asked us for to match list 1 with list 2. Under list 1 you have the scientific names, under list 2 you have your common names, you have to match it correctly. Okay, so Rhizopus you've been studying from lower grades, even in your school you may have studied. Rhizopus is the common bread mold, so A will be matched with 3. Eustilago is the smut fungus, 2. Puccinia is also called the rust fungus, C can be matched with 4. Agaricus is the scientific name of common mushroom, so D can be matched with 1. So the correct answer to this question is A3, B2, C4 and D1, which is option 3. Has everyone answered this question correctly? Let me know in the comments. Has everyone answered this correctly? Now let's take a look at this question from Principles of Inheritance and Variation. The question reads, in a plant, black seed color. Black seed color is the phenotype. The genotype is, it can be homozygous dominant, capital B, or heterozygous dominant, which is capital B, lowercase b, over white seed color, which is lowercase b, lowercase b. Excuse me. In order to find out the genotype of the black seeded plant, over here, you can't really look at the genotype. You can see the phenotype. You can see that the plant produces black colored seeds. But you can't say for sure whether its genotype is capital B or capital B lowercase b. Whether it is homozygous or heterozygous, you can't say for sure by looking at the plant's phenotype. So if you have to identify whether a plant that is producing black colored seeds are homozygous or heterozygous, what kind of cross would be performed, right? That is what this question is asking us. So we must perform a test cross here. What do we perform? A test cross. What do we do in a test cross? You take this plant which has the dominant phenotype, whichever is showing the dominant phenotype. It could be capital B or capital B lowercase b. We take this plant and cross it with a recessive plant, which is lowercase b. So with this cross, you can find out whether it is homozygous dominant or heterozygous dominant. If it is homozygous dominant, then all the progeny will produce black colored seeds. If it is heterozygous dominant, you will get plants producing black colored seeds and plants producing white colored seeds in 1:1 ratio. Okay, so that is a test cross. So in order to identify its genotype, we have to cross it with a recessive plant. So the correct answer to this question is option 4, right? Now let's take a look at our next question. This again is from the chapter Principles of Inheritance and Variation. This is a question based on incomplete dominance, right? Okay, so a pink flowered snapdragon plant was crossed with a red flowered snapdragon plant. What type of phenotype is or are expected in the progeny? They've told us clearly that they're talking about the flowers in snapdragon plant. They've not given us the genotype in this question, right? We know that snapdragon plants, if we are considering the character flower color, show incomplete dominance. It produces 3 different colored flowers. They can be red or pink or white. Whenever it is homozygous, capital R, it will be red. If it is heterozygous, it will be pink. If it is homozygous recessive, it's going to be white. So if they're crossing a pink flower producing plant with a red color flower producing plant, then what kind of progeny are we going to get? From this you get just one kind of gamete, capital R. From this you will get capital R and lowercase r. So in the progeny, you will get both red colored flowers, red color flower producing plants, as well as pink color flower producing plants. So which among these is the correct option? Only pink flowered plants? No, it's incorrect. Red, pink as well as white flowered plants? No, white will not be produced. Only red, that is also incorrect. Red as well as pink flowered plants, that is the correct answer. So the correct answer to this question is option 4. Excuse me. So let's take a look at the next question. This is a question from the chapter Principles of Inheritance and Variation again. Under list 1, we have 2 or more alternative forms of a gene. When a gene has more than one alternating form, what do we call it? We call it an allele, right? So A can be paired with 3. Cross of F1 progeny with homozygous recessive parent. F1 progeny, you do not know its genotype. If you cross it with a homozygous recessive parent, what would you call such a cross? We just spoke about it in this question, right? I told you that this is a test cross. Whenever the F1 progeny is crossed with a homozygous recessive plant, it is called a test cross. So B is a test cross. Cross of F1 progeny with any of the parents, dominant or recessive. If you're going back one generation and crossing it with the parents, we call it a back cross. So C can be matched with 1. Number of chromosome sets in a plant, what do we call it? We call it ploidy, right? If there is 1 set of chromosomes, we call it haploid. If there are 2 sets, we call it diploid, 3 sets triploid, and so on. So the correct answer to this question is A3, B4, C1 and D2, which is option 1. Right? Now let's move on and take a look at our next question. This is a question from the chapter Biomolecules again. So in the question they are asking about lecithin. Okay. Lecithin is a small molecular weight organic compound found in living tissues. Is an example of what is lecithin what is lecithin an example for? Is it a glyceride? Is it a carbohydrate? Is it amino acids? Is it phospholipids? What is it? Lecithin is a type of phospholipid. We've learnt about phospholipids. Phospholipids are present in your cell membranes. Phospholipids have a phosphate head and 2 hydrocarbon tails. They are present in the form of 2 layers as a bilayer in your plasma membrane and all other membranes of your cell organelles. So one example for a type of phospholipid is lecithin. So the correct answer to this question is option 4. Right? Now let's move on and take a look at our next question from the chapter Microbes in Human Welfare. Over here, there are different microbes, the scientific names mentioned under list 1, and what are the products that we obtain from them is mentioned under list 2, and we have to match it correctly. Clostridium butylicum, it is a bacterium that is used in the commercial production of butyric acid. So A can be matched with 3. Saccharomyces cerevisiae is the scientific name for yeast. We all know that yeast or brewer's yeast is used in the production of alcoholic beverages or ethanol. So the B can be matched with 1. Trichoderma polysporum. Trichoderma polysporum is a fungus used in the commercial production of a chemical called cyclosporin A. Cyclosporin A is an immunosuppressive agent that is used in patients who have undergone organ transplant. So C can be matched with 4. Streptococcus. Streptococcus is a bacterium which we use to obtain an enzyme called streptokinase. Streptokinase is called the clot buster, right? It is used to break blood clots within our blood vessels. So the correct answer is A3B1C4D2. A3B1C4 and D2, which is option A. Now let's move on and take a look at our next question. This is a question from the chapter Anatomy of Flowering Plants. This is regarding the stomatal apparatus. In the given figure, which component has thin outer walls and highly thickened inner walls? So it is this cell, the guard cell, that covers the stomatal aperture, that has thick inner walls and thin outer walls. So over here, D represents the stomatal aperture, C represents the guard cells, B is your subsidiary cells, and A is the other epidermal cells. So it is C or the guard cells that have thin outer walls and highly thickened inner walls. So the correct answer to this question is option 3. Now let's take a look at the next question. This is a question from the chapter Morphology of Flowering Plants. Which of the following is an example for actinomorphic flowers? What are actinomorphic flowers? Actinomorphic flowers are those flowers that show radial symmetry. What does radial symmetry mean? Radial symmetry is when you take a flower, you cut it in a plane, radial plane, any radial plane that passes through the center, you're going to get two equal halves. If it is such a flower, we call it actinomorphic flowers. Among the given options, Datura is an actinomorphic flower. It is a flower belonging to the family Solanaceae. This plant belongs to the family Solanaceae. In Solanaceae, all the flowers are actinomorphic, right? Pisum, Sesbania, Cassia, all of these are not actinomorphic. They are zygomorphic. So the correct answer to this question is option 3. Let's take a look at our next question here. This is a question from the chapter Molecular Basis of Inheritance. A transcription unit in DNA is defined primarily, I suppose, by 3 regions in DNA, and these are with respect to upstream and downstream end. What are the 3 regions in a transcription unit? In a transcription unit, you will have a promoter, you will have the structural gene, and you will have a terminator. Promoter is a site or a sequence of DNA where transcription will get initiated. Terminator represents the region where transcription process will end, right? So in a transcription unit, you will have 3 components. At the 5' end of the coding strand, you will have the promoter, and then you'll have the structural gene. Towards the 3' end of the coding strand, you will have the terminator. So the 3 parts are promoter, structural gene, and terminator. So the correct answer to this question is option 2. Next question. This is a question from Biotechnology, Principles and Processes, but half of it is cut, so I don't really know what is the fate of a piece of DNA carrying. Okay, we'll get back to this question in a little while. I'll find out if I can get a clearer image of this question. Meanwhile, we'll continue with the other questions. Okay, so this is a question about auxin, a plant growth regulator from the chapter Plant Growth and Development. Auxins are used by gardeners to prepare weed-free lawns, but no damage is caused to grass as auxins. Okay, so they're saying auxin is used as a weedicide to prepare weed-free lawns by gardeners, but it is not causing any harm to grasses. Why is that so? Is it because it does not affect mature monocotyledonous plants? Is it because it can help in cell division in grasses and cause growth? Is it because it promotes apical dominance? Is it because it promotes abscission of mature leaves only? Now, if you look at the options, all of them are the physiological effects of auxins. But they are specifically asking here regarding its use as a weedicide. You know that 2,4-D is an auxin. 2,4-dichlorophenoxyacetic acid is an auxin that is used as a weedicide. We specifically use 2,4-D as a weedicide in farms where monocots are grown. this weedicide or this chemical will not, harm the monocot plants. It will only harm mature dicot plants. If in your farm or in your garden, you have grasses and monocot plants, and here and there are some dicots growing and they are not looking good, or they are causing a reduction in your yield, then you can use 2,4-D as the weedicide, because it specifically targets only mature dicot plants and will not harm any monocot plants. Okay, so the reason why auxins are used by gardeners as weedicides in lawns is because it does not affect mature monocotyledonous plants. So the correct answer to this question is option 1. Yes, Karamjeet Kaur, every question in the botany section at least is from the NCERT textbook itself. Yeah, Karamjeet, after finishing the discussion of this, I'll tell you what are the books that you can buy. Okay. Let's take a look at the next question, which is from the chapter Biomolecules. In the chapter Biomolecules, you learn about enzymes, and after enzymes, you have something about cofactors. So this is a question from that portion of the chapter. The cofactor of the enzyme carboxypeptidase is flavin, heme, zinc, niacin. For the enzyme carboxypeptidase, a metal ion acts as a cofactor, and that metal ion is zinc. So the correct answer to this question is option 3. Alright, now let's move on to our next question. This is a question from the chapter Molecular Basis of Inheritance based on lac-operon concept. The lactose present in the growth medium of bacteria is transported to the cell by the action of permease, polymerase, beta-galactosidase, acetylase. You know, in the lac-operon, you have 3 structural genes, LAC-Z, LAC-Y, LAC-A. LAC-Z produces beta-galactosidase, LAC-Y produces permease, LAC-A produces trans-acetylase. If in the medium there are bacteria growing and in this medium there is lactose, if the lactose has to enter the bacterial cells, which is the enzyme that will help in picking up lactose from the surrounding medium, the enzyme is permease. Option 1 is the correct answer. Beta-galactosidase helps in breaking down lactose into glucose and galactose. Trans-acetylase helps in trans-acetylation reaction during the metabolism of lactose. But specifically, if the lactose present in the surrounding medium has to be transported into the bacterial cell, the enzyme that is required, or the enzyme that helps transport lactose into the cell, is permease. Alright? So the correct answer to this question is option 1. Now let's take a look at the next question. This is a question from the chapter Principles of Inheritance and Variation. Which one of the following can be explained on the basis of Mendel's law of dominance? Okay, so there are 5 statements given to us. We have to identify which one is true for Mendel's law of dominance. Mendel proposed the law of dominance based on his monohybrid cross results, right? So let's read the first statement. Out of one pair of factors, one is dominant and the other is recessive, right? Whenever there is heterozygous condition, whenever there is, a dissimilar pair of alleles, then only one of them will express itself. That is called the dominant allele, and the one that does not express itself is called the recessive allele, right? So out of one pair of factors, one is dominant, the other is recessive. This is true if it is heterozygous condition, right? Now let's take a look at statement B. Alleles do not show any expression and both the characters appear as such in the F2 generation. Alleles do not show any expression. Alleles do express themselves. It's wrong to say that they do not show any expression, and both the characters appear as such in F2 generation. Of course, they appear in the F2 generation, but alleles do show expressions. That is incorrect. Factors occur in pairs in normal diploid plants. Yes, in all diploid organisms, factors do occur in pairs. So this is correct. The discrete unit controlling a particular character is called a factor. We call it alleles and genes today, but back then Mendel called it factor. So this is a correct statement. the expression of only one of the parental characters is found in a monohybrid cross. That is true. In the F1 generation, only one of the two characters are expressed. Only one of the two traits are expressed. So that is correct. So among the given options, only A, C, D, E are correct. In all the other options, you have B. So option 4 is the correct answer. Alright. Now let's take a look at our next question. This is a statement based question from the chapter Biotechnology and its applications. Statement 1 reads, Bt toxins are insect group specific and are coded by gene Cry1AC. This is a statement taken as it is from NCERT. Bacillus thuringiensis is a bacterium. This bacterium has certain genes such as Cry1AC. These genes produce proteins that are insecticidal, that can kill the insects. Okay, there are different genes that targets different specific insect groups. That is what statement 1 says. So this is the correct statement. Statement 2, it is given that Bt toxin exists as inactive protoxin in Bacillus thuringiensis, which is the bacterium. However, after ingestion by the insect, the inactive protoxin gets converted into its active form due to the acidic pH of the insect gut. Okay, so what basically happens is, whenever you introduce these Bt Cry genes into plants by biotechnology, or if you take these Bt spores and spray it on plants, if an insect is feeding on the leaves of that plant or feeding on some part of that plant, if the insect ingests that, the Bt toxin that is present in its inactive form will get activated inside the gut of the insect because the insect gut is alkaline in nature. It does not have acidic pH, it has alkaline pH. The alkaline pH will activate the toxin, and as a result, it will cause formation of holes in the gut of the insect, and as a result of that, the insect will die. Okay, so what is given in statement 2 is correct with respect to protoxin being inactive. After the insect ingests it, it becomes activated, but it does not become activated because of the acidic pH of the gut. It becomes activated because of the alkaline pH of the gut. So statement 2 is incorrect. Okay, so over here statement 1 is correct, but statement 2 is false. So the correct answer to this question is option 1. Now let's move on and take a look at our next question, which is also statement based from the chapter Anatomy of Flowering Plants. In statement 1, it is given that parenchyma is living but collenchyma is dead. Parenchyma is living, yes, but collenchyma is not dead. Collenchyma also is living. That makes statement 1 incorrect. Collenchyma, if you know, is also called the living mechanical tissue, right? So collenchyma is not dead. That makes statement 1 incorrect. Statement 2 reads, gymnosperms lack xylem vessels, but presence of xylem vessels is the characteristic of angiosperms. Gymnosperms, yes, majority of gymnosperms lack xylem vessels. Transport of water majorly happens not through vessels, it happens through tracheids in gymnosperms. So yes, majority of gymnosperms lack xylem vessels, and xylem vessels is a characteristic feature of angiosperms. Yes, that is the reason why angiosperms are called angiosperms. You know, blood vessels are related to angio, right? Angiogram and all of that. Angio means vessels. Because angiosperms have vessels, we call them angiosperms. So statement 2 is correct. Gymnosperms lack xylem vessels, but presence of xylem vessels is a characteristic of angiosperms. This is a correct statement. Statement 1 is false, but statement 2 is true. So the correct answer to this question is option 2. Let's take, excuse me, a look at the next question, which is a statement based question again from the chapter Cell Cycle and Cell Division. There are 2 statements given. Both these statements are regarding prophase 1 of meiosis. Let's see what the first statement reads. Chromosomes become gradually visible under light microscope during leptotene stage. leptothene is the first stage of prophase 1 of meiosis. During leptothene, condensation of DNA would have begun. So you can see chromosomes becoming more and more compact during leptothene. So it has started to become visible. This is a correct statement. The beginning of diplothene stage is recognized by dissolution of synaptonemal complex. This also is a true statement. Diplothene is the penultimate phase in prophase 1 of meiosis. It happens just before diakinesis. So what happens then is, after crossing over, during crossing over, the homologous chromosomes would have come opposite each other, paired, formed a bivalent or a tetrad. There is a synaptonemal complex formed. After crossing over has happened, the two chromosomes have to move apart. They have to move away from each other. Then the synaptonemal complex has to dissolve. So that dissolution of synaptonemal complex begins in diplothene, right? So the synaptonemal complex begins to dissociate, and the chromosomes will remain attached only at sites of crossing over, which we call chiasmata. All of these are features of the diplothene stage. So over here, statement 1, statement 2, both are correct. So the correct answer to this question is, option 3, both statement 1 and statement 2 are true. Okay. Now let's look at the next question. This is a match the following type of question. Under list 1, you have different cell organelles, and under list 2, you have their functions, and we are required to match it correctly. Nucleolus. What is nucleolus? Nucleolus is a region present inside the nucleus in eukaryotic cells. It's not bound by any membrane, and it's kind of takes up a lot of stain and appears like a dark spot under the microscope. What exactly is happening at the nucleolus? In the nucleolus, there's a lot of rRNA being synthesized. So A can be paired with 3. What is centriole? Centriole is a structure, it is an organelle that you find only in animal cells, and it has the organization like the cartwheel. It will have 9+0 arrangement of microtubules. So B can be paired with 2. B is 2. What are leucoplasts? Leucoplasts are a type of plastids that you find only in plant cells. Leucoplasts are colorless, they do not have any pigments. Their major function is to store food, right? So they store foods either as proteins, lipids, or carbohydrates. So leucoplasts 4. Golgi apparatus. Golgi apparatus is where glycosylation will take place. Glycolipids, glycoproteins, they are formed in your Golgi apparatus. So that is the site of formation of glycolipid. D can be matched with 1. So the correct answer to this question is A3, B2, C4, D1. A3, B2, C4, D1. Option 3 is the correct answer. Now let's look at the next question. This is a question from the chapter Biodiversity and Conservation. List of endangered species was released by FOAM, IUCN, GEAC, WWF. So it was released by IUCN, which is the short form or the abbreviation for International Union for Conservation of Nature and Natural Resources. So that is what will publish the Red Data book, where you have a list of the status of different species, whether they are endangered, whether they are extinct, whether they are vulnerable, and all of that. Okay. So IUCN, option 2 is the correct answer. Next, we have come to the section B of botany. We finished answering 35 questions. The next question is from the chapter Cell, the Unit of Life. The DNA present in chloroplast is, we all know that chloroplast is a double membrane bound organelle, and within the chloroplast you find DNA, right? What is the nature of that DNA? Is it linear or circular? Is it single-stranded or double-stranded? The DNA that you find inside a chloroplast is very similar to the plasmids that you find in bacterial cells. So it is going to be circular and double-stranded. Okay, so the correct answer to this question is option 4. Just 1 minute. Next question. Which of the following are fused in somatic hybridization involving 2 varieties of plants? If there are 2 varieties of plants that have to be fused in somatic hybridization, what are we going to use for fusion? Somatic hybridization, generally when two cells are fusing, it is the gametes that fuse. The male gamete will fuse with the female gamete, right? But in somatic hybridization, what happens? Somatic cells will be fused. Okay, so what do we use in somatic hybridization? We use protoplast. Generally in plants, you have a cell wall. Within the cell wall, you have the cell membrane. Cell membrane and all of the contents inside, this is the protoplast. So you take whichever plant variety you want, you get rid of the cell wall, and then you take two protoplasts and fuse the protoplast. What you get is a somatic hybrid. Okay, so the correct answer here is option 1. Next question, identify the correct description about the given figure. This is a diagram from your NCERT textbook itself. So let's see what the options are. Cleistogamous flowers show autogamy, compact inflorescence showing complete autogamy, wind pollinated plant inflorescence showing flowers with well exposed stamens, water pollinated flowers showing stamens with mucilaginous covering. This is an inflorescence of a wind pollinated flower, possibly a grass, where you can see that the stamens are exposed. Because the stamens are exposed, the pollen grains, when the anther dehisces and disperses the pollen grains, they can be released easily. Okay, so this is a diagram of wind pollinated flowers. So option 3 is the correct answer to this question. Now let's take a look at the next question, question number 139. This is a question from the chapter Plant Growth and Development. Spraying sugarcane crop with which of the following plant growth regulators increases the length of the stem, thus increasing the yield? So in sugarcane plant, you know that the stem is of economic importance because it is from the stem that you get sugar, right? So if you have to increase the yield, then increasing the stem length or the internodal length will directly cause an increase in the yield, right? So if there is any hormone or a plant growth regulator that can do the job, that can increase the internodal length of the sugarcane stem, which one is it? Is it cytokinin? Is it abscisic acid? Is it auxin? Is it gibberellin? The correct answer is option 4. Gibberellins or gibberellic acids, they are known to increase the internodal length of sugarcane stems, as a result cause an increase in the yield. Okay, now let's look at our next question. This is a question from the chapter Molecular Basis of Inheritance. Under list 1 you have names of different scientists, and under list 2 you have their the experiments that they performed or what their contribution is. So you have to match it correctly. Okay. Frederick Griffith performed the transformation experiment using Streptococcus pneumoniae and mice. So A can be paired with 3. B. Francis Jacob and Jake Monod, they elucidated the lac operons. So B can be paired with 4. Hargobind Khurana is involved in identifying the codons. What codon codes for which amino acid? Deciphering the genetic code. So C is paired with 1. Michelson and Stahl performed the experiment using E. coli bacteria and heavier isotope of nitrogen to identify or conclude that replication of DNA happens semi-conservatively. So D is 2. Correct answer to this question is A3, B4, C1, D2. A3, B4, C1, D2. Next question from the chapter Biomolecules. Match list 1 with list 2. Excuse me. So there is a little small table at the bottom right corner where you have different proteins mentioned and what their function is. This question is taken from there. A similar question appeared like 2 or 3 years ago also in NEET exam. So GLUT4 is a protein that enables transport of glucose from the surrounding medium into the cells. So A can be paired with 4. Insulin, as you all know from the chapter chemical coordination and integration, insulin is a hormone. It is a protein hormone. So B is paired with 1. It regulates the blood sugar levels. Trypsin. About trypsin you would have learnt in the chapter digestion and absorption. Trypsin is an enzyme that helps in breakdown of proteins. So trypsin is an enzyme. C is paired with 2. Collagen. intercellular ground substance that you see in connective tissue. So D is paired with 3. Correct answer to this question is A4, B1, C2, D3. Option 3 is the correct answer. Alright, now let's take a look at the next question. Given below are two statements. Excuse me, I'll be back in just a bit. I'm sorry, I have a sore throat. Yes, I'm right here, Noorjahan. Excuse me. So this is a question from the chapter photosynthesis. The question reads, given below are two statements. In statement 1, it is given that in C3 plants, some oxygen binds rubisco. Hence, carbon dioxide fixation is decreased. That is true. Whenever rubisco binds to oxygen, photorespiration will take place. Whatever fixed carbon is there, that will also be lost. So, carbon dioxide fixation definitely will be decreased in C3 plants if rubisco performs oxygenase activity. What is given in statement 2? In C4 plants, mesophyll cells, I think this is show very little photorespiration, while bundle sheath cells do not show photorespiration. Okay. So in C4 plants, mesophyll cells show very little photorespiration, while bundle sheath cells do not show photorespiration. This is a wrong statement because in mesophyll cells there is no rubisco at all. In C4 plants, mesophyll cells lack rubisco. If rubisco itself is not present, then how can it perform oxygenase activity and how can photorespiration take place, right? So in the absence of rubisco, photorespiration will not take place at all. In C4 plants, rubisco is concentrated only in your bundle sheath cells. It's completely absent in your mesophyll cells. So it will not show any photorespiration. Both bundle sheath cells and mesophyll cells will not show any photorespiration. So over here statement 1 is true and statement 2 is false. So the correct answer is option 1. Let's take a look at the next question. Identify the step in tricarboxylic acid cycle or TCA cycle or Krebs cycle which does not involve oxidation of substrate. In which of the following steps oxidation is not happening? That is what we are required to find out. Okay. So in the step where succinyl CoA gets converted to succinic acid, is there oxidation happening? Is it a step where oxidative decarboxylation is taking place? Over here, there is no oxidation taking place. When isocitrate is converted to alpha ketoglutarate, oxidation happens. Malic acid is oxidized to oxaloacetic acid. Here also oxidation takes place. Succinic acid to malic acid conversion. Here also oxidation takes place. Where oxidation does not take place is what we are required to identify. And the correct answer here is option 1. Let's take a look at the last but one question. No, yeah, we still have 6 more questions. This is question number 144, a match the following type of question. Very easy question from the chapter Respiration in Plants. Under list 1, you have citric acid cycle, glycolysis, electron transport system and proton gradient. Let's see and match it correctly. Where does citric acid cycle take place? Citric acid cycle is our Krebs cycle. Krebs cycle takes place in the mitochondrial matrix, right? The site of Krebs cycle is mitochondrial matrix, so A is paired with 2. Glycolysis. Where does glycolysis occur? Glycolysis occurs in aerobic, anaerobic respiration, both in the cytosol or the cytoplasm itself. So B can be paired with 1. Electron transport system. Where are the complexes required for electron transport system present? They are present in the inner mitochondrial membrane, right? So C can be matched with 4. Next, proton gradient. Proton gradient is, there is a gradient that exists, proton gradient that exists on either sides of the inner mitochondrial membrane. On one side of the inner mitochondrial membrane, towards the inner side you have the matrix, towards the outer side you have the intermembrane space. So these protons get accumulated in the intermembrane space, and as they move through the complex 5 or the ATP synthase complex, ATP will get synthesized, right? So this proton gradient is present across the inner membrane. Proton gets accumulated in the intermembrane space of mitochondria. So D can be paired with 3. Correct answer for this question is A2, B1, C4, D3. A2, B1, C4 and D3. So the correct answer is option 4. Now let's take a look at the next question. This is from the chapter Molecular Basis of Inheritance. This is a statement based question again. In the 4 options, 4 statements are given. We are required to identify which is the correct one. Which of the following statements is correct regarding the process of replication in E. coli? Basically prokaryotic replication. In option 1, it is given DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' as well as 3' to 5' direction. This is incorrect. It catalyzes only in 5' to 3' direction. In option 2, it is given that DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' direction. This is the correct statement. In option 3, it is given that DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 3' to 5'. This is incorrect. Option 4, DNA dependent RNA polymerase catalyzes polymerization in one direction, that is 5' to 3'. If you look at this statement as a standalone statement, it is correct. But look at the name of the enzyme. It is not DNA dependent DNA polymerase, it is DNA dependent RNA polymerase. Okay. So DNA dependent RNA polymerase is involved in transcription, not replication. Okay. So in the question you can see they are talking about replication. In replication, DNA dependent DNA polymerase is involved. This as a statement is correct, but it is correct with respect to transcription and not replication. So the correct answer to this question is option T, option 2. Alright, now let's take a look at the next question. In an ecosystem, if the net primary productivity NPP of the first trophic level is 10x kilocalories per meter square per year, what would be the GPP of the third trophic level of the same ecosystem? We all know that primary productivity is about plants, right? So in primary productivity, you have two types. You have net primary productivity and then you have gross primary productivity. What is gross primary productivity now? Gross primary productivity is the rate at which total biomass is produced by plants by the process of photosynthesis. Not all the biomass that plants produce by photosynthesis will be available for the consumers. Some of it the plant will use for its own respiratory needs, right? So whatever biomass has been synthesized, some of it will be used by the plant itself. So some of it will be used by the plant itself, that is represented by R, which is the respiratory loss. Whatever remains is the net primary productivity. This net primary productivity will be available to the consumers. Okay, so to the second trophic level, which is your herbivorous organisms or plants or animals that feed on plants, net primary productivity will be available. So, if an ecosystem, the NPP of plant is, how much is it? 100x kilocalories per meter square per year, then all of it will be available to the second trophic level. Now, in the question they have asked, how much will be available to the third trophic level? Over here, we have to apply the 10% law. In one trophic level, when energy is being transferred from one trophic level to the next trophic level, you know that only 10% will be transferred. The remaining 90% is generally lost as heat, right? So, if in the second trophic level, you have 100, then only 10% of it will go to the third trophic level. 10% of 100 is 10, right? So, the correct answer to this question is option 1, 10x kilocalories per meter square per year. This is a slightly tricky question. They have mentioned net primary productivity of the first trophic level. We know for sure that net primary productivity is whatever is available to the consumers. So, all of the net primary productivity, 10x kilocalories per meter square per year, will be available to the second trophic level, which is your primary consumers. So, 10% of that will be transferred to the third level, which is your secondary consumers, right? So, 10% of 100 is 10. So, the correct answer here is option 1. Now, let's take a look at the next question, which is a match the following type of question from the chapter Morphology of Flowering Plants. So, there are a couple of examples given under list 1, and what are the examples for is given under list 2. We have to match it correctly. Rose is given. Rose is an example for perigynous flower, right? So, A can be paired with 2. P is an example for marginal placentation. All marginal placentation, B is 4. Cotton is an example for twisted aestivation. C can be paired with 1. Mango is an example for a drupe. Drupe is a type of fruit which has a stony endosperm. So, D can be paired with 3. Correct answer here is A2, B4, C1, D3, which is option 3. Now, let's take a look at the next question from the chapter Biodiversity and Conservation. This is a match the following type of question, very simple question. What is Robert May known for? What is his contribution? He has estimated that the global species diversity is around 7 million, right? So, A is paired with 3. Alexander von Humboldt is known for species area relationship. After exploring many jungles, he said that as you increase the area explored, the species richness is also going to increase up until one point, right? So, Alexander von Humboldt, species area relationship. Paul Ehrlich explained the importance of species diversity by using the analogy in Rivet-Popper hypothesis. So C is paired with 4. David Tillman performed long-term ecosystem experiments using outdoor plots to help us understand how important species diversity is with respect to stability in an ecosystem. So D is paired with 2. Correct answer is A3B1C4D2. A3B1C4D2. Option 4 is the correct answer. Here is our next question. This is a question from the chapter Morphology of Flowering Plants. It is a match the following type of question where under list 1 you have different types of stamens and under list 2 there are examples for it. We have to match it correctly. Monoadelphy is a condition in which all the filaments in a stamen are fused to form one bundle while the anthers are free, right? Monoadelphous example is China rose or hibiscus. A is paired with 4. Diadelphous is when there is fusion in the stamens or in the filaments of stamens where they are forming two bundles. This can be seen in peas where you have totally 10 stamens of which 9 are fused and 1 is separate. This is like two bundles, right? B is 2. Polyadelphous condition where you have many bundles formed, that can be seen in citrus plants. So C can be paired with 1. Epiphyllous condition is where the stamens are fused with the tepals or the perianth components. So that can be seen in lily plant, which is a monocot. So D is 3. A4, B2, C1, D3. A4, B2, C1, D3. Option 3 is the correct answer. Now let's take a look at the last question in the botany section. This is a statement-based question. Read the following statements and choose the set of correct statements. This is from the chapter Plant Kingdom about Phaeophyceae or brown algae. There are 5 statements given. We have to find out which ones are correct with respect to Phaeophyceae members or brown algae. Okay. So in statement 1, it is given that asexual reproduction occurs by biflagellate zoospores. That is true. In brown algae, you have zoospores that have 2 flagella, and it is by means of these biflagellated zoospores that asexual reproduction takes place. In statement B, it is given that sexual reproduction is by oogamous method only. This is not true. It may be isogamous or anisogamous or oogamous. Okay, so this is incorrect. Stored food in the form of carbohydrates, which is either mannitol or laminarin. This is true. Statement D, the major pigments found are chlorophyll A, C, carotenoids and xanthophyll. This is true. Vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. This is also true. So among the given 5 statements, A, C, D and E are true. So the correct answer to this question is option 1. So with this we have covered the entire botany section. Following this, we will be discussing questions from the other sections as well. Stay tuned, all right? So hopefully all of you have done really well in your NEET exams. Stay tuned for the other subjects. Okay, I'll see you very soon. Bye.
Hey champions, hello, hello, hello students, are you getting my voice? Are you getting my voice? Is everything fine? Yes, I guess we are live. So guys, how was your paper? So hope your paper went well. Now it's time to discuss zoology. So all my students are ready. So if you look at the paper, so first let's talk about the syllabus. You know, there were a lot of changes as per the NMC. Some topics they were added which were not there in the latest NCRT. So people, the students, they were very much confused. And not only you, in fact, we were all, also confused of what kind of paper are we going to get this time. So if we look at the paper was very, very, very easy. All questions, they were direct and everything we have discussed in the various series. So if I talk about my latest series, which was our NEET 50 champions, and in that series, we discussed everything, whatever was there in the paper. So I'm super happy. So hope all my students, they are also excited to know about the answers of the zoology part. So let's not waste any time and let's start the session. Okay, so this was the first question. Here, buddies, I am starting. This is 1 second. This is your T3. Right here, buddies, I'm talking about the T3. So in the T3, this was the first question, question number 151. So in the 151 question, there were two lists. So majorly the questions, they were either the match the following, assertion reason type of question, or the statement based type of questions. So here you can have a look. So here the two lists are was given, the list 1 and the list 2, and we were supposed to match. So first is the typhoid. So typhoid, you know, that is a salmonella. It is caused by salmonella typhimurium, which is a bacteria. So here, what is this? This is a bacterial disease. For the A, the correct answer is 4. I will not go. See, by looking at just one option, you can get the correct answer. Isn't it great? Yes. So here, if we talk about the leishmaniasis, this is because of the Leishmania donovani, which is a protozoa. Next is the ringworm. Ringworm is a fungus, microsporum. Etc. All these three we have already discussed. Filariasis is by the nematode, which is Wucheria bancrofti. Yes. Now, moving on to the next question, question number 152. These are the two lists, again, the most favorite type of questions. Now, the first one here, the list 1 and the list 2, two lists were given. First is a non-medicated IUDs. Non-medicated one, we talked about the 3 type of intrauterine devices. And the non-medicated one we have is the Lippes loop. Copper releasing is multi-load 375. Now, Lippes loop, multi-load 375, these are the copper releasing. Hormone releasing is your LNG 20 and the progestasert. Implants are the progestogens. For the A, correct answer is 3. See, we got the answer. These two can be correct. B, it is the 1. For the C, it is the 4. So we got our answer. Option 2 is correct. Yes, easy peasy. Now, let's move on to the next one, 153. The presence and absence of hymen is not reliable indicator of virginity. This line is there in the NCERT. Yes, this is not an indicator of virginity. The statement is 100% shat pratishat correct. Now, next one is the hymen is torn during the first coitus only. See, this only always spoils the answer. So, this is not the correct answer. Basically, because of the sports or bicycle riding, there are so many reasons. Because of our day-to-day activities, this hymen can break. So this is not the correct answer. So this is the correct one. This is not the correct statement. 1 is true and statement 2 is false. So we are going with the option number 1 in this case. Yes. Now next one. In both the sexes of cockroach, a pair of jointed filamentous structure, which is anal cerci. And you remember many times, if I talk about the PYQs, we always talk about the anal style present in the male cockroach. And that is actually creates a sexual dimorphism between a male cockroach and female cockroach. But here they talked about the anal cerci, which is present in male as well as a female cockroach. And I discussed, if you remember, we discussed the cockroach also because it was there in an MC syllabus, though it was not there in the latest NCERT. And see, here the correct answer is a 10th segment. Yes, correct. Okay. Now moving on to the next question, 155. Here again the two columns were given. Now here the first list 1, list 2. Let's first talk about the list 2. Provide additional space for neuron. Regulate posture and balance. Posture and balance, the first thing that comes in everyone's mind, that is the cerebellum. So for this the correct answer is the cerebellum. That means for the D, correct answer is 1. See, we got our answer. So this is how you can determine the answer within a short duration of time. But definitely we have to explore the other, things also. Controls the respiration and the gastric secretion. So what is the one which controls? It's the medulla. Medulla. Vomiting center, where it is present? In the medulla. You remember? Now, connects different region of the brain. Yes, it is a pons which connects a different region of the brain. Neurosecretory cells are present in the hypothalamus, which are endocrine, which secretes hormone, as well as it controls the various activities of the body. So for the A, the correct answer is 3. B, it is a 4. We are going with option number 4. Now, moving on to the next is, which is not a steroid hormone. This is steroid hormone, this is steroid hormone, steroid hormone. This is not the steroid hormone. Yes? Okay. Next one. Which of the following is correct product of DNA-dependent RNA polymerase to the given template? Now, many students, they did this mistake. Now here if I talk about the DNA dependent RNA polymerase, so here we are talking about the RNA polymerase. So RNA polymerase will do what? It will help in the formation of the RNA from the DNA. So we have a sequence of DNA given over here. Can you see? Sequence of DNA. So from the sequence of DNA we have to form a RNA. So here we are talking about the process of transcription. So here if you look at the sequence will be A, right? A opposite you will get U, G, then A opposite you will get U, then A, then you will get C, C, G, A, U, U, U. So like this you have to do. So if you'll match this, you will get the answer A, U, G, U, A. A, U, G, U, A. This can be correct. This can also be correct. C, C, G, C, C, G, U, U, U. This is not the correct. This is the one. Option 1 is the correct one. So like this you can match and you can find the correct answer. Now here, these are the question number 158. Very easy, direct question from your notes, and in fact this was a question from the tissues. Yes, isn't it? So if you look at the tissues, 3 tissues diagram was given. The first, second and the third one. Second one clearly indicate it is a smooth muscle. So what is this? This is a smooth muscles. Right? Present in the gut, right? Major part of the gut we have the smooth muscle. In the uterus we have is a smooth muscle. If I talk about the first one is a skeletal muscle, here branching are there, so here we are talking about the cardiac muscle. So for this question, what is the correct answer? Correct answer is skeletal, triceps, stomach, cardiac. This is the correct answer. Was that easy? Yes. Can we move further? Now, following are the stages of cell division. Now here different stages are given. Very easy question. So here we have to arrange. So first if I talk about the cell cycles always start with the G1 phase. So first we have is a G1 phase. Then we have is the, see this one, synthesis phase, S phase. Then we have is a G2 phase. Then what happens is a nucleus divides. So this will be the number 4 and this will be the number 5. So this is the E, then C. So this is the correct answer, C. So this the particular set which I am discussing, the questions are same in various sets. It's just that there is a reshuffling. There's a reshuffling of options also. So you have to explore, you have to calculate your marks wisely. Moving on to the next is, which of the following are autoimmune disorders? Most appropriate you have to find out. Okay. So myasthenia gravis, autoimmune rheumatoid arthritis, autoimmune, this is autoimmune disorder, this is muscular, this is not, gout is also not. Here the correct answer is A, B, E. Correct answer is option number 4. Was that easy? Yes. So here also the correct answer is option number 4. Now, list 1 and 2, again 2 lists were given. So let's have a look at the correct answer. So first is a lipase, and here some bonds are given, which these, these enzymes, they break. So first lipase. So lipase which helps in the digestion of fat. So in the fat you will find the ester bonds are there. You remember the ester bond? In the triglycerides we talked about in the chapter biomolecule. So this is the correct answer. Next is the nuclease, the phosphodiester, protease, protein, definitely it will break down the protein. So in the protein we have the peptide bond. Now amylase, glycosidic bond. So for the first, the correct answer is 2. See, from the first itself we got the correct answer. See. So these were the questions which were from the biology. They were very easy, they were very direct, and they require very less attention, very less time. So isn't it great? So you can invest, like if I talk about this paper, I know many of students, they would have invested better time in physics and chemistry because there the numericals are there. And in this part, the biology, whole biology was super easy. Lines were direct from your NCERT. The flippers of penguins and dolphins, your birds and the mammals. What is the relation? They do not belong to the same category. They are the different categories, isn't it? So here, these are the type of the convergent evolution. So these are the convergent, 2 different converging. Convergent evolution, right? So these are the what? These are the analogous structures. Now, next one. Again, list 1 and 2 were given and we have to match the correct one. So first is given, first let's talk about the expiratory reserve volume. After expiration, whatever volume is there, so that is the expiratory for the reserve volume. So next is a tidal volume and the inspiratory reserve volume. So all these are what? This indicates that we are living. And what are these? This is our vital capacity. When we are expiring, when we are inhaling, exhaling, what is this? We are living. So this, we call it vital capacity. So this is the correct answer. So for the C, the correct option is the 1. See, we got our answer. So like this, you can match the other options also. Okay, now tidal volume, normal, 500 ml, which we take and we take out. Expiratory reserve volume, that will be the expiratory capacity. Normally, tidal volume, expiration. So this is my expiratory capacity. I'm taking out 500 plus some expiratory volume also, expiratory capacity. Next is a tidal volume, inspiratory reserve volume, my inspiratory capacity. Expiratory reserve volume or residual volume, both will work in my body and they will be the one which will be involved in the gases exchange. So these are the functional residual volume. We got our answer. Which of the following factor will not affect the Hardy-Weinberg equilibrium? Hardy-Weinberg equilibrium, basically it is that equilibrium where the gene frequency tend to remain constant in a randomly mating population where there is no migration, there is no recombination, no mutation. So here, if I talk about this, if gene migration is there, definitely Hardy-Weinberg equilibrium will be affected because some new genes will be added, some genes might be deleted. Constant gene pool, this will not affect. This will support that equilibrium. So this is the incorrect. So genetic recombination, this can affect, right? The genetic drift, it will also affect. So all these are the direct from the chapter evolution. Now next one, given below some stages of human evolution, arrange them in the correct sequence. Okay, correct sequence. Past to recent. Yes, so they have given something which is recent. Okay, so if I talk about the modern man, so who's a modern man? Modern man we have is a Homo sapiens. So Homo sapiens, if I talk about this, that means these two options, they are 100% shat pratishat correct. And for this, the first organism that came was a Homo habilis, then the Homo erectus, then the Neanderthals, and then came the Homo sapiens. So this is at the number 4. So look at this. Let's see what is the correct answer. We have is the first Homo habilis, A. This is the correct answer. Okay. Now, next one. Following are the stages of pathway for the conduction of an action potential through the heart. So action potential always is generated by SA node. Majorly it is generated by the SA node. So from SA node, this potential goes to the AV node. Right? Then to the AV bundle, right? To the bundle of his atrioventricular bundle of his, isn't it? So let's have a look. So it is clearly given over here. So first of all, we have is a SA node. So that means E is a correct answer. That means these two options, they are 100% correct. SA node say directly it goes to the AV node. So here it is the AV node. So here E, then comes the A. Then comes the A. Yes, SA node to the, AV node, sorry, to AV node, this is EC. Okay, then comes the AV bundle, that is a bundle of his. Then there are some branches of this bundle, number 4, and then to the ventricular wall, that is through the Purkinje fibers. Okay, so it is the E, then comes the C, then comes the A. This is the correct answer. Was this question direct from your notes? Absolutely right, guys. So next one, moving on to next. Which of the following factors are favorable for the formation of oxyhemoglobin in alveoli? Here, if I talk about the hemoglobin binding to the oxygen, it depends on various factors. One of the factors is low carbon dioxide concentration. So the carbon dioxide concentration should be less, oxygen should be more. When carbon dioxide is less, so that means H plus ion concentration is less. That means pH will also be affected, right? And here if I talk about this, here the temperature will be more as compared to the tissues where the metabolic reactions are occurring. So here the correct answer is high oxygen and lesser H plus ion concentration, right? So pH is also more. Now, here some match the following like a funda was given. Very easy again. Okay? And if you know some very easy points which I have discussed with you multiple time in sessions. Arey, please focus on this. Question will definitely come. See, now the question is here. And if I talk about the enzyme replacement therapy, what is this? Enzyme. We are replacing. And you know, this word is mentioned in the gene therapy. Gene therapy, where we are replacing the gene. So this is the correct answer. For the D, correct answer is 2. C. So these were some free marks, if I talk about some free marks. And if you know the little concepts, so by that means you must have qualified the NEET. So here, this is the correct answer. So we'll go for this option, but definitely we will explore other options also. And alpha-1 antitrypsin, that is used for the treatment of emphysema. Then if I talk about the cry1AB, that is for the corn borer. AC is the bollworm. Yes, this is the correct answer. 169. Okay, so 2 other statement-wise question, 1 assertion and 1 reason. Let's look at the correct answer. So first was the assertion given. FSH act upon the ovarian follicles in female. FSH act on the ovarian follicle, and due to which ovarian follicles they grow. Primary, secondary, tertiary, girlfriend. That means the Graafian follicle. Leydig cells in male. FSH act on Leydig cells. FSH act on Leydig cells? No. LH, L for LH, and LH act on the Leydig cell. So this is wrong. Assertion is wrong. Now let's look at the reason. Growing ovarian follicles secrete estrogen in female, while interstitial cells, right, the outside cell, the Leydig cells, they secrete androgen. Androgen ka one example we have is a testosterone. Absolutely 100% statement is correct. So this is wrong, this is right. Okay, assertion is true, but assertion is false, but reason is true. I will go with this option. So moving on to the next is, here one diagram is given, and multiple times I have discussed in session by students, everyone. Just imprint this diagram in your mind. So here 2 different X and Y we were supposed to identify, and many people, many students, they feel problem, like they feel like if we talk about the restriction site, so they fail to identify different restriction site. But the only 2 things which they remember is Ori and rope. Everything is given, only Ori and rope was not there. So this is the Ori sequence. X is the Ori sequence and Y is the rope sequence. And Ori determine the copy number. So gene X is responsible for controlling the copy number, and the Y involved in the replication of protein. ROP, replication protein. You remember the proteins which are involved in the replication. So very beautiful. This question was easy and super happy. So match, again a match the following was given. I don't know why many students did this mistake. First, cocaine. And this is the super important topic, cocaine. Erythroxylum coca. A is the correct 3 answer. So this is the correct answer. See, from the first itself, cocaine. Many times we discuss in human health and diseases that this topic is super important. And cocaine, erythroxylum coca, and we got our answer. So next one is the heroin. So heroin we basically take out from papaver somniferum. Morphine. This is marijuana is from cannabis sativa. Morphine is a painkiller, sedative in the surgery. Yes, so we got our answer. This is the correct one. Now, next one. Again, some statement was given and we have to find out the correct one from the option. Annelids are true coelomates. Yes, given in NCERT. Yes, bio was easy. Bio was super easy, Kale. Now here, this is 100% correct. Poriferans are pseudo. They don't have mesoderm. Then how can they say they are pseudo-coelomate? Pseudo, false. Ask. We ask those peoples again and again. So this is Asclepianthus. Acoelomate? No. Asclepianthus are pseudo-coelomate. Platyhelminthes acoelomate. Now, next one. Given below are the 2 statements. In the nephron, the descending limb of loop of Henle is impermeable to water. I guess this question I have discussed minimum 7 to 8 times if I talk about the NEET 50 days challenge in that series. And if you have seen that session, if you have seen those series, I know my students, they have done great. Like, I'm proud of them because this is direct from there. And this is not the correct answer. Descending limb of loop of Henle, only water comes out. Ascending limb of loop of Henle, Na comes out. So this is not the correct answer. PCT, columnar, cuboidal. It's not the intestine. Incorrect. So here, both the statements, they are incorrect. Both the statements, they are... No. Again, match the following. The first we have is the fibrous joint. Fibrous, immovable joint. Where it is present in the skull. Do not allow the movement. A, correct answer is 3. A, correct answer is 3. Fibrous joint. Now next is the cartilaginous joint. Vertebrae. Here, intervertebral disc. Absolutely right. Next, hinge joint. Hinge. This is hinge joint. Hinge. In Hindi, in hinge, we call it kabza. This is a kabza joint. Hinge joint. So this hinge joint is the knee also, and this elbow joint is the same. Ball and socket. See, this is a ball and socket. So this is the correct answer. Humerus and pectoral girdle. Similarly, here your femur and the pelvic girdle. Same. So here the correct answer is this. Moving on to the next. Which of the following is not a natural? Not a natural is a walled diaphragms. They are not the natural. Rest all are natural way of contraception. Match the following list. Again, list 1 and list 2 was given. Pleurobrachia, tenoplana. Pleurobrachia comes under the tenophora. Radula, pila radula, stomochord, characteristic feature of the hemichordate. Air bladder, ostick thighs, bony fishes, they are heavy, so God has given them air bladder so that they can float easily in the water. A, correct answer is 2. See, this can be correct answer. Okay, so the B, it is the 1. B, it is the 1. For the C, it is a 4, and D is a 1. We'll go with the option number this. Now, next one. Exoneme, again match the following. Exoneme, cilia flagella, cartwheel, centriole, cristae, inner wall of mitochondria, fold, satellite chromosomes. This is the correct answer. For the A, it is a 2. Can be correct answer. Cartwheel, B, it is the 1. We got our answer. Now, next one. Which of the following is incorrect? Incorrect. Bioreactors are used to produce small scale. No. Bioreactors, they are used to produce the large scale bacterial culture for the 1000 liters. If you remember that this data is there in the NCERT, 1000 liter. So we got our answer. This is incorrect. So we will go for this option because incorrect was asked. Bioreactor agitator system, right. Optimum growth, right. Stirring type, absolutely right. So this is not the correct answer. So we'll go with the option number 1. So this is incorrect. It is not for the small scale. It is for the large scale. Okay, again from the botany, like obviously there's a cell division, cell cycle and cell division. This was a question. So different stages are there. So first, if I talk about the stage, first stage is a leptotene stage. So for the leptotene, it is a chromosome look like a fine thread. So for the D, the correct answer is 3. Yes. Okay. Then comes, after the leptotene, we have is a zygotene. Zygotene, there is a synaptonemal complex formation. For the C, correct answer is 1. We got our answer, C. Then comes the pecitine, okay, and then comes the diakinesis. Leptotene, zygotene, then comes the pecitine. For the pecitine, this is the reappearance of the recombination nodule. Then comes the diakinesis, the completion of terminalization of the chiasmata. Correct answer is option number 1. Now next one, very easy question. Common cold. It's a rhinovirus. Next is hemazoin, plasmodium. For the A, it is a 3. We got our answer. See, if you know about the common cold, you have the answer. You remember Vidal test, it is for the typhoid. For the C, it is the 2. So there are 2 options, but we'll go for this option. Allergy, people can get allergy because of the dust, because of the mites, because of any food thing, which can be peanuts, which can be, mushrooms, etc. So again, assertion, reason type of question. One is labeled as assertion and other is labeled as reason. Breastfeeding during the initial period of infant is recommended by doctor for bringing a healthy baby. Is this statement correct? 100%. So breastfeeding is important. Because, and you remember how did we discuss this? We discussed this by keeping a word over here, because colostrum contains several antibodies absolutely essential to develop resistance for the newborn baby. This is right, because colostrum has antibodies. What is that? That is IgA. So this is the correct explanation. We are going with option number 3 in this case. Yes, it was super easy, guys. Super easy. Again, all these are direct from NCERT. All these examples, they are direct from NCERT. Pterophyllum is your angelfish. Myxine is your hagfish. Pristis, sawfish. Exocetus, flying fish. It is the 3. A is 3. B is 1. C is 2. D is 4. Yes, clear? Okay. Now, 1 second. Yes. Okay. TI plasmid, T, tumor. I means inducing. So, it induces the tumor. This is correct. Now, next one. Which of the following is not a component of fallopian tube? Infundibulum is there, ampulla is there. There is a isthmus is also there. Fundus, name itself is there. Uterus. So it is there in uterus. It's not there in the fallopian tube. So here option 3 is correct. Again, a super easy question. Now here the 2 lists were given. If I talk about the Down syndrome is a trisomy of chromosome number 21. It is a correct answer is 3. See, we got our answer. Many students, they were confused between the alpha thalassemia and beta thalassemia, but the examiner, the NTA, they gave you a cheat here. And what was that? If you know the Down syndrome, which is there in your NCERT, you will be able to solve all of them. Now, Klinefelter is your XSY syndrome. So this is the correct answer. So we will go with the option number 1 in this case. Next, which of the following statement about the non-chordates was given? Pharynx is perforated by gill slits. These are the chordate character. Notochord is absent, non-chordate character. Central nervous system dorsal. We have central nervous system dorsal. So this is a chordate character. Heart is dorsal. We have a ventral heart. Heart is dorsal is a non-chordate character. Post, this is also a non-chordate character. So B, D, E. This is the non-chordates characters. Chalo. Agla. Now, if I talk about the next question, here the next question is related to the chapter evolution. Now, if I talk about the first one, the amongst these era, what comes first is the Pterozoic era. So, Pterozoic era, it is the option number 1. For the B, it is the 1. Invertebrates came over here. This is the 1, this is the 1, this is the correct. Okay. Then comes the Cenozoic era. Cenozoic era was that era where the mammals came. Oh, after this, definitely there are others also. Then came some Mesozoic. Mesozoic was for the fish and once again, no, there's a mistake. So Pterozoic is absolutely fine. Clear? Pterozoic is absolutely fine. But the Mesozoic is for the birds and reptiles. Cenozoic was for mammals. Paleozoic was for the fish and amphibian. Here the correct answer is option number 2. Done? Clear? Yes. Now we are right. Now next one. The cerebral hemispheres are connected by nerve tract. 2 cerebral hemispheres, they are connected by the nerve tract, which is termed as corpus callosum. Given in NCERT, 100% correct. Now the statement 2, brain stem consists of medulla oblongata, pons, cerebrum, cerebral hemisphere. No. So statement 1 is correct and statement 2 is incorrect. Very beautiful question. Yes. Now, moving on to the next is here again hormonal control was given. GnRH, this is a hormonal control of spermatogenesis was there. GnRH, so GnRH will helps in the anterior pituitary to secrete LH and FSH. So A is your FSH. Okay. Then L for LH, L always act on L. That is a Leydig cell. So B is your Leydig cell. We got our answer. Leydig cell. Then FSH act on Sertoli cells, which secrete some factor which helps in the process of spermiogenesis. So here D is a spermiogenesis. We are going with option number 3. Now, match the list 1 and 2. Again, your botany question. So, if I talk about here. Now, first thing is, for the termination of transcription, we have is a row factor. Row dependent termination, you remember? So, the B, it is the 3. We got our answer. Right? RNA polymerase 3. RNA polymerase 3, we have is a snRNA and tRNA. Now, next comes the splicing of exon. Small nucleoribonucleoproteins helps in the splicing. Tata box is a promoter. Tart. You remember? So, here the correct answer is option number 2. So, if you remember, I made a chart also for different diseases there in the chemical coordination and integration. Exophthalmic goiter, eyes are protruding outside. This is because of hypersecretion of the thyroid hormone, protruding eyeballs. Exophthalmic goiter, Graves' disease. Acromegaly, adult में. So, excessive secretion of the growth hormone. Cushing syndrome related to adrenal. Creatinism, we are creating a baby. In the baby, when there is very less production of the thyroid hormone, whatever baby will create, that will be mentally retarded and stunted growth. This, for the A, correct answer is 3. These two are correct. For the B, correct answer is 4. Again correct. For the C, it is the 1. We'll go with option number 2. Okay. Again, list 1 and 2 from the topic, your structure, organization in animals. Again, see, 2 questions from tissues, right? Deleted from NCRT, but this is there in the NMC syllabus. So how can we skip? Unicellular gland epithelium, goblet cells of alimentary canal. Now, next one, compound epithelium. Okay, salivary gland, first of all, multicellular glands. Yes. Then comes a pancreas, endocrine epithelium, secretes hormone. Islets of Langerhans, acini cells are also there, but the islets of Langerhans are also there. Okay. Then comes the compound epithelium, moist surface of buccal cavity. Here compound epithelium are there so that it can protect us from the abrasion, whatever food we eat. So for the 1, correct answer is 3. So we got our answer, see. Now, moving on to the next is 2 statement. Bone marrow is a main lymphoid organ where all the cells, yes, bone marrow, your WBCs, RBCs formation occur. Both bone marrow and thymus provide microenvironment for the development. Development of T lymphocyte occur in bone marrow. Maturation occur in the thymus. So both are important. Both are important. This is right. This is also right. This is also right. Statement 1 and 2 both are correct. Now, here comes the next one. Again, see, look at this. The structure used for storing of the food, storage, cockroach, crop. Ring of 6 to 8 blind tubule, foregut and midgut, helps in digestion. It is the hepatic key. Now, 150 colored junction of midgut and hindgut. It is a Malpighian tubule. Structure used for grinding, todo fodo. That is a gizzard, mastication of food. For the A, it is a 4. For the B, it is the 2. We got our answer. For the C, it is the 3. For the D, it is the 1. For this question number, option 3 is correct. Now, choose the correct statement from the given below. Juxtamedullary nephron. Corticals, they are present on the cortex area. Juxtamedullary deep down. This is the correct answer. Run deep down in the medulla. Now, P wave and QRS T wave. P wave depolarization of atria. For the A, correct answer is 3. We got our answer. And for the, then comes a QRS complex. That is a depolarization of ventricle. B, it is the 2. For the T wave, yes, this is the 1. And what is a TP? That is a heart muscles are electrically silent. We got our answer. Next. As per the ABO blood grouping system, the blood group of a father is B positive. Father is B positive and mother is A positive. The child is O positive. Child is O positive, that means child is this. So that means both the parents, they are heterozygous. So this can be, this will be in fact this one. This will be IA and small I. And that is the reason whenever there will be cross, small I will come over here and this small I will also come over here. So here the correct answer is this one. So here only A is the correct answer. Very easy question. Now next one, given below are the two statements. Gauss competitive exclusion principle states that two closely related species competing for same resources. So this is not the correct answer. Same resources. Statement 2, according to Gauss principle, during competition the inferior one will be eliminated. This may be true if resources are limiting. Yes, this is correct. So here option 1 is incorrect and option 2 is correct. Yes, this is the correct. Option 1 is false, statement is true. Now, next one. Regarding the catalytic activity of enzyme, select the correct sequence. Last two questions are left. I won't take much time. We will wind up. The first thing is substrate will bind to the active site. Substrate binding to the active site. Once substrate bind to the active site, there will be this complex formation. Okay. Chemical bond will be broken down. Right? Then there will be release of product. Free enzyme now can bind to the other substrate. Option number 5. Very beautiful question. E. Then comes the A. EA. We got our answer. EA. Then comes the number, this one. Then comes the C. Then comes the B. Yes. Next one. Given below are two statements. Mitochondria and chloroplast are double membrane bound organelles. Yes. Right. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. As compared to chloroplast. So this statement is, so there is a confusion. In fact, I will definitely go that the statement is incorrect. So statement 1 is correct and statement 2 is incorrect. We are going with the option number 1 in this case. Yes, so these were the zoology questions which were very easy and direct. Hope all my students enjoyed the session. And now do not go anywhere. We will be doing the solution of the physics and chemistry. So hope your paper went well. Take care. Stay here, guys. Don't go anywhere.
Hello बच्चों, सत श्री अकाल and namaste from my side. So first of all, right, physics paper was damn easy. No question exists that wasn't discussed in Yaqeen English 2024 batch, the droppers batch, right? So there exists no question which wasn't discussed in Yaqeen 2024 batch, and many questions were from revision part. Approximately for a month revision went on, right, and we have solved more than 18 to 1900 of questions within that, right? So paper was super easy. So rapidly we are going to discuss the solution for this. We are now going to discuss the T3. Okay, so the first question, tightly wound 100 turns. See, number of turns are given, the radius of a coil is given, magnitude of the current is going, permeability value is given. So directly we are going to replace magnetic field is mu 0 I whole divided by 2R. Okay, this is for the single turn. For the n turns, you are going to multiply it by n. Okay, so now what we have to find the magnitude of the magnetic field. Okay, so by replacing those values, we will get 4.4 Tesla. Right, you have to just replace the values and get the answer. Okay, moving forward to the second question. Match the column. This time I think in this paper lots of match the columns were there, right? I can understand, but it's okay. But this is a very simple thing. For, see, we have to match the susceptibility values of the para, dia, ferro, non-magnetic materials with one another. See, if I'm talking about the diamagnetic materials, their susceptibility is less than 1, right? Ferromagnetic materials are having the susceptibility greater than, okay, ferromagnetic materials are having the susceptibility greater than, right? So here I think your answer is directly going to be 1 is going to be, right? Okay, so here the third option is going to be the perfect answer for this specific question, right? Okay, so directly moving forward to the next part. thermodynamic system is taken A, B, C, D, A. The work done, see, BC. BC volume is constant, isochoric process, work done is 0. Okay, volume is constant, isochoric process, work done is 0. Right? Isochoric process, work done is 0. Now, fourth question, an unpolarized light strikes the surface at the Brewster's angle. We all know about the Brewster's law, right? The reflected light is a completely polarized, right? And refracted light is a partially polarized, right? Partially polarized. So here, the second is the perfect answer for this specific question. Let's move on to the question number 5. The ideal transformer. See, ideal transformer, in case of ideal transformer, input power that is equal to the output power. So Vs Is that is equal to Vp Ip, right? Okay. So if you all remember the number of the turns ratio, okay, that would be equivalent to Vp by Vs. So we want to find the ratio of Vs by Vp. So that will be 2:1. Okay. Direct formula based question. No here and there, right? No use discussions, nothing. Okay. So let's move on to the question number 6. The logic circuit. Okay. See, logic circuit that provides the output Y as per following. You can see the binary values in A. You can see the binary values in B. Okay. If you are going to see the final output 0, reverse is 1, 1 reverse is 0, 0 reverse is 1, 1 is 0. So here I think B bar is going to be the perfect answer for this specific question. Right? B bar. Moving forward to the 7. In a vernier calipers, n+1 division of the vernier scale coincides with n division of the main scale. So here they are saying n+1 of VSD, that is n of MSD. So direct 1 VSD, that is equal to n by n+1 of MSD, right? So least count formula is 1 MSD minus 1 VSD, replacing here value. So least count is going to be 1 by n+1 MSD. 1 MSD value is going to be 0.1 mm. So your least count in centimeters, it is going to be 100 of n+1. Okay, this is the perfect answer for this specific question. Direct formula based question. Okay, no here and there and each and every question was discussed, right? Maximum elongation into the elastic limit is given, Young's modulus is given, Hooke's law, stress, strain, right? So directly you can say strain is equal to sigma by Y, and you can compare strain to delta L by L, right? So from there you will get the 4 mm as the perfect answer. Question number 9, a horizontal force of 10 Newton is applied as shown into the figure. The masses are given. Directly draw the free body diagram. This is 2 kg, this is 3 kg, this is its net acceleration, force of 10 Newton. First of all, net acceleration is equal to net force by mass of system. So the net acceleration is going to be 2 meter per second square. Now we need to calculate the force that 2 kg applies on 3 kg. Okay. So that force that is equal to N is equal to 3A, and your answer will be 6 Newton. Right? Your answer will be 6 Newton. If monochromatic source of light is in YDSE experiment, generally we used to, right, have white light, and instead of that, suppose we are using the monochromatic light, then what happens? Right? Thousands of times it has been discussed. If we are replacing it by the monochromatic light, the central becomes bright, and nearby to that we will be having some few clutter fringes, right? Okay, so there will be the central bright white fringe surrounded by the flue. So that is the perfect answer for this specific question, right? The graph which shows the variation of the kinetic energy. If I am talking about the equation of the de Broglie wavelength, then lambda is inversely proportional to under root of E. So lambda square is in, okay, so 1 by lambda square is directly proportional to E. So if we are seeing, then Y is directly proportional to X. So we are having the graph of straight line. The nature of the graph is straight line. Moving forward to question number 12, array, balancing situation, right? Your answer is 2. Circuit is balanced. The left part of the circuit is balanced to the right part. Symmetry. You can solve it by symmetry. You can solve it by Wheatstone, everything, right? 13 number question. Okay, see, there are two solenoids, 1 and 2. Here the bar magnet is taken in this side. If I am talking about the south pole, the magnetic field always terminates to the south pole. And if the magnet is moving towards the right side, it means magnetic field is increasing towards right, left, and flux is increasing towards the left. So according to the Lenz's law, we need to oppose it, right? Okay, so here the direction of the current will be according to the Lenz's law DC, and here it will be AB. Okay, you have to just, if the value of the magnetic field is increasing, decrease it, because increase in magnetic field will give you increase in flux, right? Okay, consider the following statements, A and B, identify the correct answers, right? Okay, see, for the solar cell and for the reverse, see, PN junction diode never works into the reverse bias. So I think here the majority charge carrier isn't going to be the issue. So here PN junction diode never works into the reverse bias. For solar cell, the IV characterized graphs live into the fourth quadrant. Yes, okay. A is correct, B is incorrect. PN junction diode can't work in reverse bias. As of that reason, okay, it is highly doped, and here comes the Zener diode that works actually into the reverse bias. Okay, let's move on to the question number 15. Oh, 2 times we have to apply the Snell's law. A light ray incident, okay, at some specific angle, it travels through the prism. Okay, see, this is R1, this is R2. You can say R1 plus R2. Here it is 90 degree, that is a prism angle. Suppose if I am going to apply the Snell's law at P, so that is 1 into sine of 30, that is eta into sine of R1. Okay, and if I am going to apply the, let's say here Q, Snell's law at Q, so it is eta into sine of, here it is R1. So here we can write 90 minus R1, that is equal to 1 into sine of 90. So from this you will get root 5 by 2, okay, as the refractive index of the prism. That is what we need to find out. Okay, let's move on to the question number 16. Given below are the two statements. The potential at the axial point. See, if you want to find the potential at the axial point, it is Kp cos theta whole divided by R square. I think in the reason it is 2. No, at the axial point it is not. Okay, so assertion is right and reason is wrong. Okay, so in 16 question, assertion is right and reason is wrong. Okay, let's move on to the question number 17. The moment of inertia of the thin rod about an axis passing through the midpoint perpendicular. Okay, see, this is a rod. Okay, from the mid, the moment of inertia is 2400. So the formula for the mid, that is equal to 2400. Replace all of those values, right? You will get length approximately at 8.5 centimeter. Okay, let's move on to the question number 18. Supply voltage is given. You need to find the terminal voltage. So current, that is equal to E by R plus R, right? So that is equal to 10 by 5. The current flowing is 2 ampere. So the terminal voltage V is equal to E minus. Okay, so your answer is 8 voltage. Question number 18 is solved. Let's move on to the question number 19. Very easy. See, you can go through the formula, you can have the transition, right? Delta E, that is equal to Hc by lambda. But if you are going to calculate, right, through this much transitions, right, it is a very time consuming process. So instead of that, less is the transition, high is the wavelength. 1, 2, 3. Okay, lower is the transition, high is the wavelength. So I think in question number 19, yeah, 4 is the perfect answer for the 1 is going to match 3. That's it. Okay. Let's move on to the question number 20. If c is the velocity of the light into the free space, then correct. Okay. The energy of the photon E is equal to hv. The velocity of the photon is c. The momentum is correct. In a photon, okay, A, B, C, D both are correct. This is wrong. Photon does not basically contains the positive charge, right? Okay, so A, B, C, D all are correct in question number 20. Very nice. Here alpha particle is released, so 290 will become 286, 82 will become 80, right? Okay, so here again one positron is released, so it is 286, that will be 79, right? 286, 80, 286, 81. This is your perfect answer, right? Let's move on to the question number 22. The displacement is given by this. See, displacement is given 2t minus 1. Force is given 5 Newton. Differentiating this equation with respect to time, you will get velocity, that is 2 meter per second. If you want to calculate the power, that is Fv, 10 watt. Right? The third is the perfect answer for this specific question. Right? Okay? Third is the perfect answer for the right specific question. Let's move on to the question number 23. Okay. See, the output of the given logic gate is basically your answer is going to be the AND gate only. Right? Okay? Your answer is going to be AND gate only. Okay, but if you want to follow the truth table, see here A and B both are same. So you can say A dot A bar, B dot B bar. So this is Y1, this is Y2. A B plus B bar. Okay, so here Y1 dot Y2. Okay, from this you will get the answer. Okay, the AND gate, the final answer would be right, the AND gate. Okay, this will be plus, right? The final answer is going to be right. Okay, the AND gate. Let's move on to the question number 24. The mass of the earth is 1 by 10. Gravity on the planet, G on the planet, that is equal to G mass on the planet by R P square. Mass on the earth by R E square. Okay, this is 1 by 10 into 4. So approximately your answer is going to be 3.92. Okay. So gravitational acceleration on the planet is earth into 0.4. If you are going to replace the value of G as 9.8, you will get your answer, right? Okay. Given below are the two statements, electrons are electrically neutral, equal number of positive and negative charges, correct. Atoms are stable, they emit the characteristics, yes, they are also correct, right? Everything is correct. Okay. Statement 1 and statement 2 both are correct. Okay. The wheel of the bullock cart is moving on the road as shown into the figure. If it is a linear, speed is v. Which of the following options are? Okay. See, here speed is 2v. Here speed v minus v is 0. So P moves faster as compared to Q. Because here the relative speed is 2v and at Q the relative speed is 0. Okay. A particle wave moving with the uniform speed. See, velocity and acceleration both are the right. In a circular path, the direction of the velocity is also changing, right? And the acceleration also. Okay, so both are the vector quantity. So the change in the direction into the vector quantity is also a change. So both are varying. 28. A thin flat circular disc of radius 4.5 is gently kept on the surface of water. Surface tension is this. So how much excess force is required? Surface tension is force per unit length. Force is equal to surface tension into the one free surface 2 pi r. Right? So replacing those values, you will get 19.8 millinewton as the perfect answer for the specific question. Okay? 29. The uniform magnetic field. Okay, in a uniform magnetic field, a magnetic needle performs 20 oscillations in 5 seconds. Moment of inertia is given. Magnetic moment magnitude is given. 20 oscillations, 5 seconds. Right? One oscillation, 0.4 second. So the time period is t is equal to 2 pi under root of I by mb. Replacing 0.4 by 2 pi is equal to under root of I by mb. Right? So it is 0.4 into 0.4 whole divided by 4 pi square is equal to I by mb. So replacing all of those values, you will get this as a perfect answer for this specific question. Right? Two bodies A of same mass undergoes inelastic collision. Right? One is moving. Okay. Directly you can apply the conservation of linear momentum. Okay. As it is a completely inelastic collision, so after collision they will stick. So both will be having the common velocity. So V1 by V2 ratio is going to be 2:1. That's it. Okay. Conservation of linear momentum, net force is 0. If you are going to compare the standard equation, this is the amplitude, this is omega. So 2 pi by t is equal to pi, t is equal to 2 seconds. So pi and 2 is the perfect answer. The quantities which have the same dimensions of the solid angle, strain and angle. Okay, a thin spherical cell. See, if it is thin spherical cell, charge inside is 0, electric field is 0. If electric field is 0, potential is constant, right? So at C and P, potential is same. So potential difference is 0. Okay, a bob is whirled horizontally. Okay, by means of a string. Okay, suppose if it is whirled horizontally, so T is equal to directly proportional to, right, omega square. So T1 by T2, omega 1 square by omega 2 square. From this you will get 4T as the perfect answer, right? Moving forward to question number 35. Long wire of the 100 ohm divided into 10 parts, right? So each wire will be 10 ohm. 1, 2, 3, 4, 5. Again 1, 2, 3, 4, 5. Okay. Each will be 10, 10, 10, 10, 10. Okay. So this resultant would be 10 by 5, 2, 10, 10, 10, 10, 10. This will be 50. Final resultant is 50 plus 2, 52. Okay. Section B, the following graph represent an ideal gas at the pressure compared with those of the Charles law. Charles law, PV is equal to nRT, right? V by T, that is equal to constant. T by V is equal to, is directly proportional to slope. It is directly proportional to pressure, right? So P1, here angles, theta 1 is greater, less than theta 2 is less than theta 3, right? So pressure P1 is higher. Parallel plate capacitor is charged through the battery. If the current in the circuit, then gap decreases, capacitance is going to get increased. Okay, gap decreases, capacitance is going to get increases. So here Q is equal to CV, E is equal to half CV square. From all of those formulas, right? Oh no, sorry. Okay, this is the next question. I'm sorry. Okay, here displacement current of the magnitude equivalent to flows. Okay, here the fourth is going to be the perfect answer, right? Here the fourth is going to be the perfect answer, right? Now let's move on to the question number 38. The property which there is only the single property. Okay, the electromagnetic waves are not originated from the charges. They are originated from the resultant oscillations of electric field and the magnetic field, right? Okay, 39. Choose the correct. See, here they have played with you all in the grammar. Can achieve, right? So you have to check the forward and the reverse bias of the diode, right? You have to check the forward and the reverse bias of the diode, right? Okay, so this is only the one. See, suppose current is flowing in this direction, so this is only convected into the forward bias. Okay, and please always take into the mind the diode always provides, right, the some amount of the barrier potential. If it is providing the barrier potential, then it is having some amount of the resistance, right? So resistance of diode plus 5 ohm, they both are connected in series. So in this case, what happens? This both resultant of this two can achieve a balance. Here they are not saying it should achieve. They are just saying it can, right? Okay. Let's move on to the question number 40. If the plates of the parallel plate of the capacitor connected to the battery, then okay, charge is stored. Okay. Okay, the plates are moved close to each other, right? Okay, that was a capacitance increases by the formula epsilon naught A by D. Okay, distance decreases, capacitance increases, Q is equal to CV. Charge energy is half CV square, right? So here the charge increases, energy stored increases. A, C, right, and E. Okay, A, C and E are correct. 41. Force is this. Alpha will be F by T square. Beta would be F by T. Okay. So if you want to balance, fourth will be the perfect answer. A metallic bar, see Young's modulus is given, alpha is given, length is given, area of cross section is given, change in temperature. The compressive force will be quite simple. Stress is equal to Young's modulus of the strain, right? Here they are talking about the thermal strain. So you can replace the value of the thermal strain into this formula to get the force, right? Okay, to get the force, right? So here 50 into 10 to the power 3 will be the perfect answer. We need to find the magnifying power of telescope, right? FO by FE, direct formula. This was discussed into the class. 28 is the perfect answer. Iron, okay, see. This is one iron bar, okay. The half of the iron bar, the length becomes half, right? And it is tilted. So the magnetic moment, the resultant, the new magnet they are talking about, okay. So if you are going to take out the resultant, it will be m by 2. Okay. 45th, a capacitor is connected to this. See, this is a VRMS value is given, right? So here if you want to calculate the IRMS value, so that will be VRMS whole divided by XC, and here it will be V0 by root 2. So here V0, that would be equivalent to root 2 into VRMS whole divided by XC. Replacing those values, you will get 0.93 ampere is a perfect answer, right? Two heaters A and B. Okay, see the power ratios are given. Power in series is 2.1 by 2 plus 1. So it is 2 by 3. Power in parallel is 3. Ratio will be 2:9. The velocity time graph is shown. See, velocity is 0, right? It means acceleration is 0. Velocity is increasing, acceleration is constant. Velocity constant, again acceleration becomes 0. Velocity decreasing, it means acceleration is constant but negative. So this is only the matching answer, right? Mass of the bob of the simple pendulum is increased to the original. T is equal to 2 pi under root of L by G, right? Okay, you will directly get your answers. The minimum energy required to launch a satellite of a mass m from the surface of the earth in circular orbit to altitude. See, this is, okay, from surface of the earth to the height 2r, right? So here you are going to apply the conservation of energy, K initial, U initial, that is K final plus U final, right? So they are asking the total energy required, okay? So first of all, from this you are going to calculate the velocity, right? At this, final kinetic energy will be 0, so G m whole divided by 3r. Okay, so when you will get the velocity and then you will calculate the total, okay, energy. So you will get 5 gmm by 6r as a perfect answer. A sheet is placed on the horizontal surface in front of this. Okay, this is the horizontal. Okay, there is one sheet that is placed on the horizontal surface. A force is needed to, right? A force is needed to. Force is needed to. Okay, to hold the sheet if it is unmagnetic. Hold the sheet if it is a non-magnetic rod. If it is magnetic, we need, yes, BIL sin theta. This much amount of the force is needed to hold. Move the sheet away from the uniform value. Yes. See, if left side and the right side forces are balanced, so there is a possibility it can move with the constant velocity, right? So here A and C is only the perfect answer for this specific question, right? So this was a very easy and the rapid solution from my side, right? Okay, so anything if you want to ask, we had asked into the doubts, right? So thank you Bacha ji, thanks a lot. This is your own physics educator Indrajit Singh signing off from the desk. Bye bye, Sat Sri Akal and Namaste from my side. Bye everyone, stay at home, stay safe, stay healthy. This is pandemic and take care. Bye.
Today here we are here for solving your NEET 2024 chemistry paper, and yes, I hope your chemistry paper is all good and you attempt all question very perfectly. Yes, paper was quite easy, yes, right? Am I right? How many marks are you expecting from this chemistry paper? It's too much easy. From your physical chemistry segment, 18 question will be asked, and please tell me how many of your chemistry questions are correct. Please tell me, all of you, how many questions you attempt in chemistry and how many of them are correct. Let we discuss. Let we start. Are you ready? Are you ready or not? Yes, I hope you are ready. Let's start. First question. For 1 mole of H2O2, H2O2O2, how many Faraday of electricity required? Right? So here we have the equation. For 1 mole H2O, we get half O2 plus twice H plus 2 electron minus. So for this, for 1 mole of H2O, we require 2 electron. So here the correct option is option number 2 for the first one. Second, MnO4 minus 2 Mn plus 2. That means we have to reduce it from oxidation number plus 7 to plus 2. That's why 5 Faraday is required. So correct option is fourth one. From 1.5 mole CaCl2. For 1 mole Ca, we need 1 mole Ca, we need 2 Faraday. So for 1.5 mole, we needed 3 Faraday. So third one, and now FeO to Fe2O3, right? So for Fe2O3, FeO to Fe2O3, we required 5 Faraday. So the correct statement is 1, and here we required 1 Faraday. So it's 3 here. So 2, 4, 1, 3 is your correct answer, and option is option number 3. 2, 4, 1, 3 is your correct answer. Done, beta? Let me move towards the next question. Next question is, which of the following is not a redox reaction? From following reaction, there is only one reaction here where oxidation number of any atom will not be changed. So it is not a redox reaction, and it is BaCl2 plus Na2SO4. It gives BaSO4 plus 2NaCl. So here we can see that, beta, for Ba plus 2, Cl's oxidation number is minus 1, Nas will be plus 1, SO4 will be minus 2, Ba plus 2 will be plus 2, SO4 will be minus 2, plus 1 and minus 1. So no change in oxidation number. No change in oxidation number. So beta, it is not redox. So the correct option is option number 2 is your correct answer. Option number 2 is your correct option. Done, beta? Next question. 1 gram of a sodium hydroxide was treated with the 25 milliliter of 0.75 molar HCl solution. Mass of a sodium hydroxide left. It's a very easy question. With 1 mole of NaOH, 1 mole of HCl is reacting to produce NaCl plus water. Now beta, for an HCl, we have volume of HCl 25 ml. So it will be divided by 1000. So it will be 0.025 liter, and its molarity given as 0.75 molar. Now beta, mass of a sodium hydroxide which is given as 1 gram. Then how many gram left? Now for this, first of all, we have to find mole for HCl. So mole of HCl is molarity into volume. So molarity is 0.75 and volume is 0.025. So here we get moles. Right now, for time saving purpose, I am using calculator. So 0.75 into 0.025. So the correct answer is 0.01875 moles. Done, beta. So they are in a stoichiometrical coefficient with each other. For both of them, the stoichiometrical coefficient is 1:1. So now moles of NaOH used is also 0.01875 moles. 1875 moles. Now moles of NaOH which is given is 1/40 mass by molar mass. So it is 0.025. Clear, beta? So now, beta, moles of NaOH which is unused is 0.025 minus 0.01875, right? So here we are solving it. 0.025 minus 0.01875. So the correct answer is 0.00625. And now if we discuss about the mass of NaOH, then it will becomes 0.0625 into 40. So it will be 0.25 gram. But here we have all option in a milligram. So it will be 10 to the power 3 gram. So into 0.25 into 10 to the power 3, which is 250 milligram. So it is 250 milligram. So your correct answer is, beta, option number 4. It is good question of stoichiometry. Right, beta? Next question. Match the following. In physical chemistry, we also notice that there are many match the following and statement 1, statement 2 types questions are asked. But they are very easy. It is a basic fundamental question of thermodynamics, which we can say that the all processes are given and we have to just match with the correct option, right? So isothermal process, the process which occurs at a constant, carried out at a constant temperature. So for A, option number 2 is correct. And yes, isothermal process, so carried out at a constant temperature. So it is a 2, option number 2 is correct option for this. And then isochoric pressure. Isochoric process is the one which carried out at a constant volume. So the third one is correct. Isobaric process which occurs at a constant pressure. So fourth one is correct. And adiabatic process, that means no change of a heat. So first one is correct. So 2, 3, 4, 1 is your correct answer and it is option number 2. 2, 3, 4, 1. Next question, beta. Activation energy of any chemical reaction. Beta, from Arrhenius equation, there are 3 questions will be asked. First question, activation energy. We all know that for activation energy finding, we have a formula like log K2 by K1 equals to EA by 2.303 R, EA divided by 2.303 R, 1 by T1 minus 1 by T2. So we have 2 different temperature, 2 different rate constant. So we can easily found activation energy by these equation. Rate constant at 2 different temperature is your correct answer. Done, beta. Now, yes, beta, but let me explain first. Each question is important. Now again match the following and it is very easy from quantum numbers. Let me start from the bottom. N is a principal quantum number. So from the principal quantum number, we can decide the size of a orbital. So correct option, size of a orbital. So second one, from angular momentum quantum number, we can decide the shape. For spin quantum number, we can decide the orientation of a spin, and from angular momentum quantum number, we can decide orientation. So correct order is 3, 4, 1, 2. 3, 4, 1, 2 is your correct answer, which is option number 4. Let me move towards the next question. It is from equilibrium constant. For the reaction Kc, equilibrium constant is given. At certain time, we have a concentration of all. Then with this help of this, we can easily find Qc. What is Qc? It is basically reaction quotient, and it is value of B into D, B into C divided by A to the power 2. So by help of this equation, we can find that 2 into 10 to the power minus 3 into 2 into 10 to the power minus 3 divided by 2 into 10 to the power minus 3 is whole square. And here we get Qc equals to 1, and now we have the Kc. Kc is 4 into 10 to the power minus 3. So Qc is greater than Kc. So reaction definitely move towards the backward direction. So correct answer is reaction has tendency to give backward reaction. Done beta. Qc is here, Qc is greater than Kc, so reaction must be going in the backward direction. Again, question from your favorite topic, Arrhenius equation, and where we can find that K equals to A into e to the power minus Ea by RT. And if we taking log ln K equals to ln A minus Ea by RT, so the slope is negative. If we compare it with the y equals to mx plus c, your slope is negative and it is minus Ea by R. These all three graphs slope are positive. So the correct answer is this, because here we get slope as a negative. Done beta. Next MCQ. Which of the following reaction for Kp is not equals to Kc? See, where Kp is not equals to Kc, where delta Ng becomes non-equals to 0, then only Kp and Kc are not the same. If delta Ng, which is moles of a gaseous product minus moles of a gaseous reactant, if it is equals to 0, then you can say that your Kp and Kc's value are same. But for non-equal to 0, see, for first reaction there are 2 moles of reactant and 2 moles of product. So it is not correct answer. For second, again 2, not correct answer. In third, we can see that there are 2 moles of product and 1 moles of reactant. So here the correct answer is option number C. Clear beta? Done. Paper was quite easy. Next question. The energy of the electron in the ground state. See, when there is a comparison between two molecules energy, you should remember only simple formula. E of He plus divided by E of Be plus 3. And for these, it's a first n equals to 1 state, and for the Be plus 3, it would be n equals to third state. Correct formula is minus 16.9 minus RH Z square upon N square. Again same, minus RH Z square upon N square. So here for He plus and for He plus we have minus x joule. So here I am putting minus x, and energy of Be plus 3 will be here. Now RH get cuts. So for He plus Z, value of Z square, helium's atomic number is 2. So value of Z square will be 4 upon its first orbital. So N will be 1. For Be plus 3, Z equals to 4. Beryllium atomic number 4. So here it comes 4 square divided by N square. N is given as 2. So 2 square. So now beta, energy of Be plus 3 will be simply 4 square will be 16. 4 cut. So 16 by 16. So here we get beta, simple equation Be plus 3 will be minus x. Clear beta? So the correct answer is beta, from here correct answer is minus x. We can simply do it by taking a ratio of z square versus n square. Done beta. So here it is a correct answer. Now let me move towards the next question. Which of the following process entropy is increased? We all know that as randomness increase, entropy increase. So in which of the following entropy increase? Liquid evaporate to vapor, entropy increase, definitely vapor forms. If temperature decrease, entropy decrease, not increase. And in next equation, solid to gas, so again entropy increase, and again molecules to atom, so entropy increase. In these three cases, A, C and D, A, C and D, in all these three cases, entropy will be increased. Next question, the highest number of a helium atom. So number of atom, we can say that for helium, number of atom, it is monoatomic. So number of atom directly proportional to moles. For all these case, which moles will be highest, number of atom will be highest. For the first case, see, for the first case, it is 4 gram of helium, so moles will be 1. For the second case, 2.27, so mole will be 2.27 liter divided by 22.7, so it is 0.1 mole. So again it is less. For the option number 3, 4 mole of helium, so here we get directly 4 mole, which is highest among all. Let me check the fourth one, 4U of a helium, that means it is only 1 atom of helium. So beta, number of helium is highest in this 4 mole, because in this option you can say that 4 into Na atoms will be present. So correct option is 4 mole of helium. Clear beta? Let me move towards the next question. Henry's law constant have a different value, so arrange them in the solubility order. See, we all know that P equals to KH into chi. So solubility is inversely proportional to KH. So as KH increase, solubility decrease. Done beta? So you can write that solubility is inversely proportional to Henry's constant. So C, 145 is the highest among all. So A's solubility will be less, and A's solubility will be least in the option number 4. So A, then the C1, because it has 35 and it is 10 to the power minus 5. So it has least value, its solubility would be highest. So correct option is option number 4 beta. Clear beta? Next question. A compound contains, for this empirical formula we have to make a table. So here 3 element is given, A, B and C. 32% of A, so 32 gram is taken, 20%, so 20 gram is taken, and remaining is a C. So 48 gram of a C will be present. Done beta? Now beta, their molar mass is given 64, 40 and 30. So their moles will be 32 by 64, 0.5. Here it will be 20 divided by 40. So 0.5, and here for last 48 divided by 32, it will become 1.5. Now taking a simple ratio. Now for simple ratio, the least number which comes is 0.5. So 0.5 divided by 0.5, it will becomes 1. 0.5 divided by 0.5, it becomes 1. And last 1.5 divided by 0.5, it becomes 3. So correct answer is ABC3 is a empirical formula. So ABC3 is your correct option. Clear beta? Next question. The plot of a osmotic temperature. See, for osmosis we have the formula pi equals to CRT. So if we draw plot of y versus t, so y versus x, so the slope is y equals to mx plus c, so the slope is CR. Value of slope is given. Sorry, we are going to plot a diagram between pi and c. So the slope is CT. Slope is RT basically, right? So if we plot diagram of pi versus C, as asked in question, the slope is 25.73. So 25.73 equals to value of R is given 0.083 and the temperature comes here is in Kelvin. Remember one thing, here many students make a mistake. Temperature equals to 25.73 divided by 0.083 and here correct answer comes which is 310, but it is Kelvin, so you have to convert that into Celsius. So 310 minus 273, so correct answer is 37 degrees Celsius. Beta, here many students make error. In hurry they write 310, but it is in Celsius, not in Kelvin. So we have to convert it in Celsius and the correct answer is 37 degrees Celsius. Next MCQ beta, the rate of a reaction quadruples, that means 4 times increase. When the temperatures change from 27 to 57, it means from 300 Kelvin to 330 Kelvin, the rate will be increased 4 times. So you can say that rate constant also increase 4 times. Now calculate the energy of activation. So we have to find the EA for this. And for this all the data is given. So again question comes from the Arrhenius, log K2 by K1 is equals to EA by 2.303 R 1 by T1 minus 1 by T2. So here log K2 by K1, that means log 4 equals to EA, which we have to find 2.303 is given. Value of R is also given in the joule, which is 8.314. Value of T1, which is 300 minus 1 by 330. So if you simplify this equation, you get answer in joule, and if you then divide it by 1000, so you get answer in kilojoule. And for this correct answer is beta 38.04 kilojoule per mole. 38.04 kilojoule per mole is your correct answer. Again, see the common things about this paper. 3 questions will be straight away asked from the Arrhenius equation. Straight away. So very good, easy paper competitively. Now this question is the heart of all chemistry paper. It is the hard question of this chemistry paper because it is the only one which is a little bit higher level question. Let me solve this. For this, consider the following reaction in the seal vessel at an equilibrium with the concentration N2, which is 3.0 into 10 to the power minus 3, O2 4.2 into 10 to the power minus 3 molar, and for NO it will be 2.8 into 10 to the power minus 3. And here they gave us that the condition at equilibrium. So here by these data, first of all we can find simply Kc. Let me find it. So Kc becomes concentration of N2, concentration of O2 divided by concentration of NO. Done. Now let me put the value here. So concentration of N2 will be 3 into 10 to the power minus 3 into 4.2 into 10 to the power minus 3 divided by, see here 2 NO. So NO square will become, so 2.8 into 10 to the power minus 3 square. And if you do the simplification, you get the answer like 1.6. Done beta. So here we have the Kc because all 10 to the power minus 3 gets cut. So now from this Kc, we have the initial concentration of NO, which is given to us separately. So we have to write the further equation against twice NO, it gives and gets N2 plus O2. And if we start with the 0.1 mole of it, initial concentration of both these two taken as 0 minus 2x plus x plus x, then it will become 0.1 minus 2x plus x plus x. And now applying Kc's formula again here to this case. So your Kc becomes x square divided by 0.1 minus 2x whole square. So 1.6 equals to x square divided by 0.1 minus 2x whole square. Now taking a square root, 1.27 nearby, x upon 0.1 minus twice x. So here by solving this, we get 0.0035 approximately. But here for this question, it is not our answer, because here for this question, we have to find dissociation constant, which is your alpha. So basically, we can write here from this, it is our C alpha. You can note that here we are doing C minus C alpha. So basically I am writing it C alpha equals to 2x. So alpha equals to 2x by C, and 2 into 0.0035 divided by 0.1. So it becomes 0.007 approximately divided by 0.1, which is 0.7 approximately. So the correct answer is option number 2. Let me correct one thing. It would be 0.035. 0.035. So 0.035 comes here also. So it's a 0.07 and then divided by 0.1. Your answer is 0.717. Done, beta. Next question. The work done during the reversible isotherm. A simple formula from your thermodynamics. W equals to minus nRT ln V final by V initial. And here we are given as a pressure. So if we convert the volume in pressure, it's a nRT ln pressure initial divided by pressure final. N is 1 mole. R is given in calorie, which is 2. And into temperature, which is given 25 degrees Celsius, so it becomes 298 Kelvin. If we convert this ln into log, it will be 2.303 log and 20 to 10. So it becomes log 2. So correct answer is minus 2 into 298 into 2.303 into log 2. Log 2's value we all know that 0.3010. So by solving this, we get the corrected answer minus 413.14 calories. So it is your correct answer. Done, beta? I hope all your physical chemistry question done. I hope so. Yes, and you get scored 80 out of 18. 18 out of 18, sorry. Yes, correct MCQ. Done. See you all next time on your own PW in English channel. Till then, bye-bye. Thank you.
Hello dear students, how are you? How was your paper? So, the last part of the paper is remaining. All your questions are solved of zoology, botany, physical chemistry, physics is done. Now we are just left with organic and inorganic chemistry. Hello everyone, good evening. How was your paper? Do let me know in the comment section. How was your paper? Yes, it was good. If I talk about chemistry, the questions were quite easy. Yes, and I don't think any tough questions except 1 or 2. So the paper was quite easy compared to last year's question paper. So let us get started with the solutions part. Yes. I hope I'm audible to you. Yes? Am I audible to you clearly? Say yes or no. Yes? Okay. So let us get started with the solution part. Intramolecular hydrogen bonding.
So if we talk about intramolecular hydrogen bonding, it is within the molecule. Yes. So if you look at 4 of the options, so if you see this option, I think it is not visible to you clearly. Let me write it. The option number C is like this. Okay, so there will be a intramolecular hydrogen bonding within the molecule between oxygen of this and hydrogen of OH. Okay, between the oxygen of nitrogen molecule, yes, and hydrogen of OH. So yes, correct answer is C. Option C will be the correct answer, dear students. Yes, option C is the correct answer. There will be intramolecular hydrogen bonding in option C. In rest all the 3, there will be a interhydrogen bonding. Okay, there you will find intermolecular hydrogen bonding, and in this case you will find intramolecular hydrogen bonding. Yes, next is filling solution A is. Okay, so we have 2 type of filling solutions, dear students, A and B. Both are given to you. Yes, this one, if I talk about alkaline solution of sodium potassium tartarate, this is your failing solution B. And if I talk about aqueous copper sulfate, you must have learned copper sulfate, but yes, again NEET is the examination of detail. How good you remember the details? So aqueous copper sulfate, so this is your A. Here most of the students should have made mistake. I think they just see copper sulfate and they take the answer. So the correct is aqueous copper sulfate. Both the failing solution A and B is given to you. We are asked about A, so C will be your correct answer, aqueous copper sulfate solution. Okay, that is CuSO4 aqueous. So this is your failing solution A. Okay, moving on to the next one. A very basic question of an organic chemistry. You are being asked about the shape of the molecules. Okay, they have asked you about the shape of the molecules. So you are asked about NH3, BRF5. It is an interhalogen compound. Yes. Next is XCF4, and next is SF6. Okay. So if we talk about ammonia, you know its structure. There is a lone pair over a nitrogen atom. Yes, and 3 NH bond like this. So this is your trigonal pyramidal. Yes, this structure is your trigonal pyramidal structure. So for A, which option is correct, dear students? For A, 1 is the correct answer. Yes, so this 3 gets ruled out on its own. Okay, but again, if you want to solve others also, we can see. For BRF5, it is square pyramidal. It is something like this. 4 fluorine atoms around a bromine and 1 F above it. So this is your square pyramidal. So this how your BRF5 looks like. If I talk about XCF4, it is your square planar. Okay, and if I talk about SF6, so it is an octahedral molecule. Yes. So you know which is the correct answer. Yes, C is the correct answer, dear students. C is the correct answer, right? You can see all the 4 geometry. Now next, the E naught value for Mn3 plus to Mn2 plus couple is more positive than that Cr3 plus Cr2 plus or Fe3 plus Fe2 plus due to a change of. Okay, so if you look closely, for manganese 3 plus and manganese 2 plus, what happens here is, one second, let me take another pen. So in the case of Mn3 plus, you get a D4 configuration. Yes, but if you go from Mn3 plus to Mn2 plus, the D4 configuration changes to D5. So it is basically going from 3D4 to 3D5. So going to 3D5, you know what it is attaining? It is attaining a half-filled configuration. Yes, so it is attaining a half fill configuration which is much more stable. Yes, you know half fill and full fill are stable. So the reason of being it more positive is that it is going from D4 to D5 configuration. Yes, so A, option number A is your correct answer, dear students. A is your correct answer. Going on to the next one. A compound with a molecular formula of C6H14 has 2 tertiary carbons. Its IUPAC name is. So all you have to do is, what you will do, you will arrange 6 carbons in a such a way that you get 2 tertiary carbons. So if I have to arrange, if I arrange 6 carbons like this, so what you can see that in this carbon and this carbon, this 2 carbon, you will get 2 tertiary carbon atoms. So if you just put hydrogen in the rest, okay. So what this molecule becomes? This becomes 2,3. It becomes 2,3 dimethyl butane, isn't it? Yes. This becomes 2,3 dimethyl butane, where you can see that there are 2 tertiary carbon is present. Tertiary, you know, a carbon which is attached to 3 other carbons, that is your tertiary carbon. So yes, option number A is your correct answer, 2,3 dimethyl butane. Moving on to the next one. Spin only magnetic moment is same for which of the following molecules? So to find mu, yes, that is your formula to find magnetic moment is root of n into n plus 1. Yes, what is n here? N is number of unpaired electrons. Yes, dear students, n is your number of unpaired electrons. So basically in all these options, wherever there will be a same number of unpaired electrons, its spin only magnetic moment will be same. Yes, as easy as that. So if you go on to find all this, you will find that here there are 4. Can you see this color? Let me change. In this case, n is 4. There are 4 unpaired electrons if you see in Cr2 plus, and in the case of Fe2 plus, there are also n is equal to 4. So B and D, yes, C is the correct answer. In the case of B and D, you can see number of unpaired electrons is 4. So n is equal to 4. Then its spin only magnetic moment will also be same. Okay. Heading on to the next one. Arrange the following elements in increasing order of electronegativity. So when it comes to electronegativity, you have to just think for phone. Yes, that is your shortcut. Phone, I am talking about F-O-N. Yes, this thing, if you just know about this simple thing, you would have answered this. So this trend is followed in which option? In only option number C. If you see this option, okay, F is more electronegative than oxygen, which is more electronegative than nitrogen, okay, then comes C and then comes silicon. So C is the correct option, dear students. Heading on to next. Which one of the following alcohols react instantaneously with Lucas reagent? Yes, so if you talk about Lucas reagent, you will see turbidity. The time of appearance of turbidity changes. Yes, we have seen that the most reactive would be tertiary, then comes secondary, and then comes primary. In the case of tertiary, the turbidity is seen within few seconds, right? So the most reactive one will be your tertiary carbon. So in the options, you have to see which one is tertiary alcohol. So option number 2 is the correct answer. Yes, this is your tertiary alcohol and it will show turbidity within few seconds when reacted with Lucas reagent. Okay, so clear till here. Heading on to the next one. 2 statements are given to you. Statement 1 says both CONH3 and COF6 complex 6 are octahedral but differ in their magnetic behavior. That is true. Both are octahedral, but yes, their magnetic behavior differs. Why they differs? I'll tell you. This one is diamagnetic while COF6 is paramagnetic. What is the reason, dear students? The reason lies in the nature of ligand. Okay, they have not asked you about the reason, but if they have to make the paper tough, the reason should be asked, and the reason is the nature of ligand. Here NH3 is a strong ligand. Okay, and what strong ligand does? It compiles molecule to pair its electrons. Okay, it will force the compound to pair up its electron. That is why in CONH3 pairing happens. Yes, pairing happens, and it makes it diamagnetic. But in the case of COF6, the nature of fluorine is weak. Yes, so it will not occur any pairing over there. So the electrons remain unpaired, which results into its paramagnetic nature. Okay, so for now both the statements are true. Yes, both the statements are true, dear students. So C is your correct answer. Okay, C is the correct answer. Clear till here? Yes. Heading on to the next one. Given below are two statements you have given with. Statement 1 says the boiling point of hydrides of group 16 elements follows the order. Okay, now these are the direct lines from your NCERT. Okay, if any of you have read NCERT with their full heart and mind, yes, they would answer it quickly. Okay, many of the lines are directly from NCERT. No mirch masala has been added. Okay, nothing was added. Yes, so statement 1 says this. It shows a trend of hydrides. Talking about statement 2, on the basis of molecular mass, H2 is expected to have a lower boiling point than the other members of the group. And what is the reason? Very basic reason, very common reason. The reason is hydrogen bonding. Yes, in water you can see hydrogen bonding. Yes, so its boiling point is the greatest among all the hydrides. Okay, so yes, both the statements are correct. Both the statements are correct, and second is the correct explanation for first one. Okay, next one. This one is the reactions from your organic chemistry. Yes, so here if you see this is nothing but the ozonolysis is being done. Okay, ozonolysis. This is a classic example of ozonolysis. Yes, so here in this fourth option would come. Yes, A4 would come. Yes. For B, it is anhydrous AlCl3 would be added to this. So for B, 1 is the correct answer. For C, what happens? See, the alcohol is converted to ketone. Yes. So which reagent would do that? CrO3. Chromium would do that. Chromium complex would do that. CrO3 chromium oxide would do that. So here 2 would come. And in this case, 3 would come. Okay, you can use either KMNO4 or KOH could be used. So the correct trend would be 4, 1, 2, 3. 4, 1, 2, 3. Yes. A is the correct answer, dear students. A will be your correct answer. Okay. Next is a compound is given to you. Okay. So what is happening over here basically is an alkene. An alkene is being converted to aldehyde. Yes or no? Yes, an alkyl is converted to aldehyde, but that is not the case. Okay, it is not possible. It is not possible for an alkene to directly get converted into aldehyde. So there is a step in between. Alkene would get converted into alcohol first. Okay, 1 degree alcohol to be specific, and then 1 degree alcohol would convert itself to aldehyde. So this is your case. For alkene to convert it into primary alcohol, BH3 would be used. Yes, and H2OH minus. Yes, anti-Markov-Nikov rule. Yes, According to anti-Markovnikov rule, you will use BH3. Peroxide effect, yes, you know the other name. The other name for anti-Markovnikov is peroxide effect also. So, in the presence of peroxide and BH3, you will get 1 degree alcohol, yes. And on the oxidation, okay, oxidation you have to use a mild oxidizing agent, okay. Mild oxidizing agent like PCC you could use. You could use a mild oxidizing agent like PCC, and your alcohol would convert itself into aldehyde. So, let us see which is the correct answer. Number 4 is the correct answer, dear students. Yes, number 4 is the correct answer. Yes. Heading on to the next one. Reagents with which glucose does not react. Okay, there is a not written. Okay, I hope you would have read this not carefully. Okay, which does not react to give corresponding test. So yeah, it reacts with Tollens reagent, it reduces it. With HCN, it forms hydrocyanine, cyanohydrin. Yes, with NH2OH, it forms oxime. So, with B and E, it does not react. So, yes, A is the correct answer. With B and E, it would not react. Yes, it would not react with B and E. Heading on to next, very easy one. Okay, very easy. It asks you about hydrocarbons. One is ethane. Okay, one is ethene, then carbon molecule, then ethyne. No need to solve everything. Yes, it is a match the following. You have to save time. So, no need to solve everything. For ethane, what happens is, they have just asked you about carbon atom. So, for ethane, 1 sigma bond. Yes. For ethene, 1 sigma, 1 pi. Yes. For C2, 2 pi. Yes, 2 pi bonds in C2 molecule, and for ethyne 1 sigma and 2 pi, 1 sigma and 2 pi. So, for A 3, for B 4 will come, for C 2 will come, and D 1 will come. So, A is your correct answer, dear students. A is the correct answer. I don't think there could be any easier question than this. Okay. This was one of the easiest question of chemistry for today's paper. Okay. Among group 16 elements, which one does not show -2 oxidation state? So, as you go down the group, what happens is metallic character. What happens to metallic character? Yes, it decreases. Yes, so -2 oxidation state. Polonium would not show. Okay. Theoretical question was there. Very easy to answer. Heading on to the next one, the boiling point of 3 isomeric pentanes follows the order. N-pentane has greater boiling point than isopentane, which is greater than neopentane. Yes, this is the correct trend. And what is the reason? The reason is, as the branching increases, okay, as the branching increases in the compound, it decreases its surface area. Yes, it covers less surface area, which means the boiling point would decrease. Yes, with a very minimal temperature it would boil. Yes, yes, the same reason is written over here, that when branching increases, the molecule attains a shape of a sphere. Yes, it piles up one over other, and it somehow takes a shape of sphere. This results a smaller surface area for contact, due to which the intermolecular forces between the spherical molecules are weak, thereby lowering the boiling point. Correct? Both the statements are correct. Okay, if you see today's paper, in the case of this statement 1 and 2, in most of the cases, both the statements were correct. Okay, the next one, the compound that will undergo SN1 reaction. Okay, so for SN1 reaction is a two-step reaction, and the reaction will depend on the stability of carbocation. Yes, and you know, for carbocation, tertiary carbocation is the most stable one, then comes primary, then comes secondary, then comes primary, and then is methyl group, right? So for SN1, tertiary carbocation is the most stable one. What is the reason? The reason is number of alpha hydrogen. Yes, that's what your hyperconjugation tells us. Yes, that's what hyperconjugation tells us. The more number of alpha hydrogen, the more stable it will be. Yes. So if you see in this case, in the option number 2, this will become a, this is your primary one, this is your secondary one, this would not form, okay. This will form your primary one and this would not form. Now, in the case of this will form a benzyl carbocation, okay, which is more stable. Yes, benzyl carbocation is more stable. So, option 2 is the correct answer, my dear students. Okay, now this is a very easy question from your practical organic chemistry. Okay, practical organic chemistry. What is said? You just have to read 2 lines. No need to read entire question. What it says, on heating some solid is converted to vapor. So what is that process in which solid is directly converted to gas? Which is this process? This question is from 9th grade. Okay, this question is from 9th grade, dear students. So the process in which solid is directly converted to vapor, that process is sublimation. Okay, no explanation required. Now this question is from your coordination chemistry. Yes, coordination compounds. If you see this compounds, okay, for A, B, C and D. So this is clearly, if I see this C option, this is your coordination isomerism, clearly visible. Yes, clearly visible. This is your solvent isomerism. Yes, this is your linkage isomerism. NO2 could link with through nitrogen also. It could link through oxygen also. So there is a 2 possibility for its linkage, making it a linkage isomerism. And this one is your ionization isomerism. So 2, 3, 4, 1 is the correct trend, dear students. 2, 3, 4, 1. 2, 3, 4, 1. C is the correct answer. Okay. C is the correct answer. Heading on to the next one. Given below are 2 statements are given to you. Aniline does not undergo Friedel-Crafts alkylation. That is true. Next is aniline cannot be prepared through Gabriel synthesis. Yes, both the statement are correct. So statement 1 and 2 are correct. 3 is the correct answer, dear students. I hope it is clear to you. I hope you are all watching this answer key. Yes, I cannot see your comments, dear students. Yes, I hope you all are watching. Yep. Arrange the following elements in increasing order of first ionization enthalpy. Again, a straightforward question from your NCERT. Okay, just a single twist, and the twist is by you exchanging beryllium and boron due to half-filled orbital. Half-filled orbital. Okay. So if you just have to exchange boron and beryllium. Okay. So fourth becomes your correct answer. The trend is same. Just you have to exchange beryllium and boron, and the reason is half-filled orbital is more stable. Fine then. The most stable carbocation, again the same question. Most stable carbocation, tertiary would be most stable, then comes secondary, then comes primary. So you just have to identify which one is the tertiary one. So yes, if you look at this carefully, okay, this is attached to, this carbocation is attached to 3 carbon. Yes, so option 2 is the correct answer. Option 2 is the correct answer. Yes, the products A and B are obtained in the following reaction respectively. Yeah, so what products you would get in place of A and B? Okay, very easy. If you don't know the reaction also, then also you can answer. How? You just have to calculate. Okay, we have learnt a very basic law, that is law of conservation of mass, which tell you that the mass of reactants and products should be same, and in order to be it the same, what would happen? The number of atoms on the reactant and product side should be same. Yes, if you know, if you have basic knowledge of this, then you can easily solve this question. See, 3 ROH plus PCl3 gives 3 RCl. So this and this has already been covered. What we are left with? We are left with 3 OH and P. So it would form H3PO3. Okay, H3PO3. Okay, I am solving it in the easiest way possible. Okay, we are not applying any chemistry over here. Okay, just applying the simplest law of conservation of mass. Heading on to the next. ROH plus PCl5. So 1 Cl has always 2 Cl, 1 and 1 means 2 Cl already been consumed. We are left with 3 Cl, 1 R is consumed. Yes, and we are left with O, we are left with P, and we are left with 3 Cl. So here you will get POCl3. Yes, you are left with POCl3. So H3PO3 and POCl3. Yes. If you don't know anything, then also this answer will go right. Okay, I can assure you of that. Okay, next, for the given reaction, okay, reactant is given to you. You have to react it with KMnO4 in H+, which means in acidic medium. Please look carefully over here that they have asked you about acidic medium, they have not asked you about basic medium. If OH minus would be written over here, your answer would flip. But now we have to think for acidic medium. Yes, in acidic medium, you will get this as 4 is the correct answer, dear students. You will get COOH over cyclohexane ring. Okay. Next, CONH3 is a homoleptic complex, where CONH3Cl2 is a heterolytic. Of course, the second statement just define its definition. In this, all the ligands are of same type. Yes, and that is known as a homoleptic complex, a complex in which all ligands are same. That is your homoleptic complex. But in the case of CONH3,4Cl2, here two different type of ligands you can see. Yes, making it a heterolytic complex. Making it a heterolytic complex, the same thing is written in statement 2. Yes, so again, both the statement are correct. Okay, statement 1 and 2 both are correct. Okay, dear students. The next one, during the preparation of more salt solution, which of the following is added to prevent hydrolysis of Fe2+ ion? So to prevent the hydrolysis of ferrous ion, you have to add dilute sulfuric acid is added. So option 2 is the correct answer. Okay. Now identify the correct answer. Let us read one by one. Dipole moment of NF3 is greater than NH3. This is wrong. Dipole moment of NH3 is greater than NF3. Okay, so this one is an incorrect statement. Next is 3 canonical forms can be drawn. That is correct. 3 resonance structure can be drawn. Not 3 but 2 resonance structure can be drawn for ozone. And BF3 has a non-zero dipole moment. No. BF3 has a zero dipole moment because fluorine is more electronegative than boron, heading into 3 different directions, making the dipole moment 0. So, option B is the correct answer. Okay. Given below are certain cations. Yes. Ma'am, I filled wrong roll number. What will happen? Let's see. I hope you could complain or do something about it. Yes, roll number is a very important thing, I guess. Okay, coming back to the solutions, dear students. arrange them in increasing group number from 0 to 6. So if you just write and arrange them in increasing group number, what you could see is you will get first CO2 plus, yes, that is your group number 2 or 0, yes, then you will get aluminium, yes, then you get CO2 plus, yes, then Ba2 plus will come, and then Mg2 plus will come, so BADCE. C is the correct answer, dear students. Okay, C is the correct answer. The next one, you have to identify the major product C. So what you are given? Iodopropane is given to you. Iodopropane is given to you. Yes, if you react it with sodium cyanide, cyanopropane is what you get. Yes, this is your A. If you do its partial hydrolysis, you will get propanol. Yes, this is your B. Yes, you will get here, wait a second, you will get something like this over here. Yes, and if you react it with NaOH Br2, so what you will get? This part will get eliminated and you will get propylamine. So the number of carbon you started with, in the end you will get same number of carbon, but instead of iodine you will get amine group. Okay, so propylamine is the correct answer. Propylamine is the correct answer, dear students. Okay, mass in gram of copper. Mass in grams of copper deposited by passing 9.6487 ampere current through a voltmeter containing copper sulfate solution for 100 seconds is, okay, so they have asked you about how much copper is being deposited. So the time is given to you 100 seconds, current is given to you 9.6487 ampere. Okay, yes, so the mass would be equal to ZIT, yes, where Z is molecular mass, yes, your molecular mass upon N factor into Faraday's constant into I into D. So what is the molecular mass, dear students? It is 63.5 of copper, right? N factor will be 2. For Faraday's constant, it is already given to you. It is 96500, but they have given you the exact one, so you have to use that. 96487 coulomb. I is given to you and 100 second is given to you. You just have to solve this. And the answer would come as 31.4 gram. Okay, please solve this. The answer would fall something like 31.5 gram. Okay, dear students. So the next question, you have two major products A and B form in the equation. So which are the major products would be formed, that you have to tell. So if you just react this reactant with PBr3, what will happen? Br will replace OH. Okay, Br will replace OH. So your A would look something like this. Here Br will attached and CH3. Okay, this is the case. Okay, so your answer is between C and D. Again, if you react in the presence of alcoholic KOH, so what happens? It removes HBr and a single bond would come over here. Okay, the single bond would form along this side, not this side, because what is the reason? The reason is the most substituted product is the more stable one, ZCF product. Yes, so according to ZCF rule, this product is more stable. So option C is the correct answer, dear students. Okay. Heading on to the next one, the 100th question, the last question, okay, of chemistry. The pair of lanthanoids ions which are diamagnetic. Okay. So for diamagnetic, the number of electrons should be even number, only then they would get paired. Okay, only then they would get paired and becomes diamagnetic. So if you see this option, in the case of CuY4 plus and ytterbium 2 plus, here the number of unpaired electrons is 0 and here the number of unpaired electron is 14. Okay, so both are diamagnetic in their nature. So option C is the correct answer. I hope it is clear to you all. Okay, then this is the complete solution of your NEET paper. I hope you have did good in the exam. So what should be your score? Please do let me know. Okay, how was the paper? How much score you are expecting? Please just tell me in the comment section. I'm waiting for you all. See, the paper is done and the solution is also done as of now. So there is no point in regretting now. Okay, there's no point in regretting. You just have to think what could be done next. Okay, so all of you, good night. You have done a great job. Just don't panic. All will be good. Okay, so good night. Take care. Stay hydrated and we'll see you soon. Bye bye.